All right, good morning, everybody. This is Allie Dunn. We're back for day 2 of CABW. We have a little bit more content to go through today. Same style as yesterday. We'll start off the morning with a panel and then a couple more sessions. And then in the afternoon, CalSFS has the poster session. So if you didn't check those out yesterday, please head over to the agenda, click the link for the Zoom meeting and check those out there. And then there's also another R workshop. So this workshop is focused more on intermediate and advanced skills. I think it's full. So if you signed up, good for you. If you didn't make it, we'll have something in the future. We also are using Slack again. So just a reminder, if you use that Slack FAQ and guide, you'll get all set up there. But use that for Q&A with the presenters, and that platform is there. So even after CABW, you can check out Slack and interact with folks there. I think that's it as far as logistics. And Angela, are you on for the poster?
Yes,
winner?
I'm here.
Hi.
Hi, can you hear me? Next slide. Awesome. Hi, so my name is Angela DePalma-Dow. I am the new president for CalSFS, California Chapter Society Freshwater Science. And I just want to say a big thank you to all the people that submitted posters, and we had one lightning talk. The competition was really fierce for the students poster competition, but everybody did a really good job with all their submissions. In total, we had 11 submissions, 7 were students, we had 2 postdocs, and we had 2 professionals submitting posters. And so for the winners, we had budgeted about $250. And so for those, we had 4 awards. We had Best Overall, which for a $100 award went to Jasmine Shen. She's an MS student at CSU Humboldt for her her lightning talk, actually, responses of macroinvertebrate drift biomass concentration to pulseful releases downstream of Lewiston Dam on the Trinity River in California. Good job, Jasmine. And then we had 3 other awards for the undergraduate category with $50. Jonathan Carmichael, he is a undergraduate at CSU Monterey Bay, with his talk or his poster, Effects of the Removal of Invasive Giant Cane on Aerial Macroinvertebrates. And our master's award was Nick Framstead from UC Davis in Turk, with his poster on nutrients from the deep, internal phosphorus loading in hyper-eutrophic Clear Lake. And then our PhD category winner was William Oda from UC Riverside, with how wastewater discharge is structuring communities in the urban Santa Ana River headwaters. So big round of applause, virtual, for everybody that submitted posters and 4 winners. Good job. And just a reminder that all those posters in the lightning talk are still available on our website. Thank you to Ryan and Hilly and Christina for putting that together. And then also thank you to all the CalSFS members and all the people that donated. Thank you for your membership and your donations. We're able to provide student poster awards like this and be able to encourage the next generation of freshwater scientists. So thank you very much. Back to you, Allie.
Cool. Thank you, Angela, and congrats to everyone. It was cool seeing everybody engage on Slack too, so good work given this remote world. Okay, so our first session up today, we have a panel, and it's focused on environmental DNA approaches for biodiversity and species monitoring. This panel is going to be moderated by Susie Thoreau with SWRCB, Southern California Coastal Water Research Project. And Susie is an ecologist at SWRCB, where she works on algal bioassessment and the use of molecular methods and biomonitoring. And she's also really been a leader for eDNA and molecular group or molecular work in California. She leads the California Molecular Methods Workgroup as well, which is a work group of the Monitoring Council. And we can put info on that in the Slack channel. And with that, Susie, I'm going to hand it off to you.
Okay, sounds great.
Okay, are we? There we go.
Okay, awesome. Thanks, everyone. I'm excited to get started today. Thanks so much for being here for our session on environmental DNA. And thanks to Allie and Shuka and Nick and Swamp and CalSFS and everyone that was involved in organizing this great meeting. So I'm Susie Thoreau. I'll be the captain for this eDNA session. And I just wanted to start us off and make sure that we are all on the same page in terms of what exactly is environmental DNA. So let's see if I can grab control here. All righty. And go to the next slide, please. Okay, awesome. So you can click through a couple times. And so as I mentioned, so for us all to be on the same page, we know that environmental DNA is simply DNA that is isolated from environmental samples. So that's in contrast to what we might think about in terms of DNA that's isolated from individual organisms like a fin clip or a cheek swab, which we don't consider environmental DNA. Often when we're talking about environmental DNA, we're talking about DNA that's in water samples. And so that could be in the form of mucus or scales or cells. But eDNA can also refer to DNA that's found in soils or sediments or biofilm samples and beyond. We like to use eDNA to target a whole menagerie of organisms, anything from bacteria to algae to invertebrates to fish and many more. So those are all potential targets for using DNA to assist them out in the environment. All right, next slide, please. Okay, you can click through. Yeah, just click through a couple. Awesome. So the next one. So why exactly would we be motivated to use eDNA-based approaches for biomonitoring and bioassessment? So I want to go over, there are a couple of key advantages and a few disadvantages that are important to keep in mind for using these eDNA-based approaches. Namely, the advantages are the fact that when we use eDNA to monitor organisms in the environment, we can do so in a really non-invasive way. Often it means simply going and collecting a water sample from the environment. We also know that these eDNA-based approaches can be really sensitive and they have the ability to detect really small concentrations of target organisms. Oftentimes, our DNA sampling and analysis can be repeatable, largely scalable, and affordable, and in certain circumstances, really quick. So it offers us a number of great opportunities to be targeting organisms that we sometimes don't have the ability to do so with traditional morphotaxonomic approaches. On the flip side, there are a number of key disadvantages that are important to keep in mind, and I think that you'll hear a number of the different speakers in today's session mentioning. One of the big ones is that we are really reliant on what we call DNA reference libraries. So these are the collections of DNA reference sequences that we like to use for identifying the organisms that are present in our samples. And right now we know that the status of these DNA reference libraries is incomplete. Not every organism we want to target has one of these reference sequences available, and that can really limit our ability to detect them out in the wild. One of the other big unknowns when it comes to eDNA-based work is the fate and transport of eDNA through different aquatic systems. So how quickly does it propagate downstream? How quickly might it degrade with different temperature or UV exposure regimes? And there are still a lot of unknowns that are currently limiting our ability to model or predict how long eDNA might persist in the environment. So that's a really active area of research. And again, something that you'll be hearing about a couple times today from the speakers in this session. All right, next slide, please. So there are a few different approaches that researchers might choose to use when doing eDNA-based monitoring. And so the first method that I just wanted to provide a brief overview of is something that you may have heard of before, and that's quantitative PCR. So when we're talking about quantitative PCR, we're talking about using the actual number of gene copies present in a sample to target your organism. One of the other methods that you'll be hearing a lot about today is what we call DNA barcode sequencing. So with DNA barcode sequencing, you're sequencing a small snippet of DNA that we call a barcode or a fingerprint, and that helps us identify the target organism in your sample by comparing it to all of those known sequences in those DNA reference libraries. Now, if you want to supersize your DNA barcode sequencing and you want to target up to thousands of different organisms all at the same time, then we do what we call DNA metabarcode sequencing, where again, you're sequencing these little snippets or barcode regions within an organism's genome, and you're using that to identify many different target organisms all at the same time. All right, next slide, please. All right, so there are a couple of key goals that I have for today's session. So first off, I want everyone to come away with a good overview of eDNA-based approaches across conservation, biomonitoring, and bioassessment applications. I'm hoping that everyone in the audience today feels like they have a good handle on the strengths and limitation of eDNA-based approaches and might even be able to identify some potential research questions that you want to answer using eDNA-based approaches. And the cherry on top would really be if you find yourself identifying potential collaborators that you see in the session that you want to contact to either join you in your research or in your monitoring program. So with that, I am going to kick it off with a bit of an overview of how we are going about advancing some of these eDNA tools for bioassessment applications here in California. So Shuka, if I could get the next slide, please. All right, and one more. All right, so right now we really are seeing a broad interest in the integration of using eDNA-based tools into routine monitoring and bioassessment applications. I think it's what's bringing us all here to this session today, is that there's real potential in integrating these different methods into these routine biomonitoring efforts. We know that there are many researchers, largely in the academic sphere, but we're seeing it more and more in the commercial space that have been using these methods with great success. So eDNA-based methods are not just a shiny new toy, but they really have the potential to be a useful one. If you go to the next slide. But what I would put forward today is that there really still is a gap that exists between what we might consider eDNA research and application. And this gap right now is inhibiting the widespread integration of molecular tools into routine monitoring efforts. So I want us to be thinking about that a lot today, and the way that we can begin to build these bridges between the researchers and the management communities to hopefully accelerate the integration of these eDNA tools into routine monitoring efforts. All right, next slide. Next slide, there we are. All right, so I would say there are three main areas that I want to focus on today that I think could help us bridge this gap between research and application. And so the first of these areas is in method standardization. So right now we find ourselves in a situation where if you are a novice user and you want to use DNA-based approaches, where do you even begin? What sampling methods should you use? And how can we ensure that if you're a novice user or an experienced user, the data that you're collecting is going to be consistent and comparable and of high quality? The second area where I feel like we could really begin to put our focus in to bridge this gap is on how we're interpreting our DNA data. So again, how are we ensuring consistent interpretation of a DNA signal? What does a positive DNA detection even mean? And how can we make sure that all the analytical techniques that researchers are using are yielding the same results, even if they're choosing slightly different methods? The third area that I'll focus on is how we communicate our DNA-based results. So right now, DNA data can seem really overwhelming and foreign. You get these really large data sets. So how can we help empower users to share their data in a way that makes it transparent and reproducible, but also in a way that our managers can use this DNA data for decision making? So these are the three areas that I'm going to focus on discussing a little bit more today. So the first one, as I mentioned, is the method standardization. So I want to start by saying that this is not a California specific problem. This is truly an international problem. In the past year or so, I've attended no less than 6 conferences that were focused on exactly this topic, on standardizing DNA-based approaches and accelerating their integration into management decisions. But this is one way that California really is unique and is a leader in that we've already begun thinking about this, and we now have a statewide work group that's focused on tackling exactly this issue. So as Ali mentioned previously, we have something called the California Molecular Methods Work Group, which was started in 2018 as a work group in the California Water Quality Monitoring Council. And what's special about this work group is, yes, they're focused on method standardization, but also it's a really diverse group of both technical experts, so academics and researchers that are using DNA methods on a regular basis, but also a number of key stakeholders in the management community and regulatory and regulated agencies that will be responsible for implementing a number of these different DNA-based tools. So by bringing together this really diverse community, we're starting the conversation now about how these novel methods can be integrated into routine applications. Next slide. So a couple of the key goals of this work group, just in case you haven't heard me talk about it before, one of the big things that we're focused on working on is developing draft standardized operating procedures for monitoring programs to be able to use as a template and implement it into their programs. Also, we'll be interfacing with stakeholder and practitioner communities as we develop these SOPs and then send them out for review for feedback, which we then integrate and make sure that these protocols are all going to be feasible for a diverse user community. And then this group will also be focused on a number of different pilot studies and intercalibration exercises once we have these SOPs in place for everyone to take for a test drive. Next slide. And so thankfully, the timing of this meeting was just so in that we have a couple of these draft SOPs now available and that are out for review. So if this is something that you want to go ahead and take a look at, you are welcome to go to the link that's on my screen now. And you can review these SOPs that we've developed and even provide feedback if you are so inspired. All right, next slide, please. All right, so the second area that I wanted to focus on was on giving our users better tools for interpreting DNA data. So the areas that I think we can really be focusing our attention is on understanding the strengths and limitation of what DNA data is telling us, and also identifying areas where we might be introducing bias due to our different analytical techniques. So once we have a suite of these standardized sample method collection, hopefully that we've been developing as part of our work group activities, how can we go about ensuring that everyone's interpreting their eDNA data consistently? So one of the ways that we wanted to partner with some of our user community in beginning to answer some of these unknowns is a pilot study that we did in collaboration with the San Diego Regional Water Board. So as part of this joint pilot study, we went into the field with a number of the water board staff. We worked through one of our sampling protocols and we developed a sampling schema to begin to better understand how eDNA is propagating through the environment. We call this our DNA tracer study, and I'll give you a little bit more of the details, but it really, I think, highlights a great opportunity of when both like the research community and the management community can come together to test drive some of these novel approaches. Next slide, please, thanks. So as part of this study, we took some marine mussels that were placed in cages in 2 different freshwater systems, and they would therefore serve as a foreign DNA tracer that then we could use to detect the propagation of DNA downstream. We used quantitative PCR, or in this case, a more sensitive version called droplet digital PCR, to detect the presence of this mussel DNA as it traveled down the stream. This was a small scale pilot study, and we were able to see, or we were looking to see if there were detectable differences in how DNA propagated in either what we consider to be an urban system, in this case, Coyote Creek in Southern California, or in a more natural system, the San Juan Creek, that we knew had a lot of organic biomass that could possibly interfere with some of our DNA signals. So if you just click next slide. So as I mentioned, we had inserted those caged mussels upstream in the stream and then monitored for them at various sampling locations, hundreds of meters downstream of where we might expect to find that DNA. Okay, next slide. And so, in fact, there were some large differences in how the DNA was propagating downstream in these 2 systems. In the urban system, which is a channelized stream, we saw sustained strong DNA signals that were able to propagate over 1,000 meters downstream. And in contrast, in our more natural system, we had consistently inconsistent or weak eDNA signals, and the signal sometimes wasn't even detectable within just a few meters of where we had inserted those caged mussels into the system. Now, this was a small scale pilot study. There are a number of different environmental factors that can be influencing how this eDNA is propagating downstream. But what we were seeing was pretty consistently in those more natural systems, a weaker DNA signal. And we attributed a lot of that to the fact that we had more organic biomatter, which is known to glom onto your eDNA and make it hard to detect in your systems. All right, next slide, please. One of the benefits of this study, as I mentioned, is because we were partnered with the water board staff, that meant that they were able to go the next sampling season and deploy some more of those same methods that we had used in our eDNA tracer study for collecting additional eDNA samples all throughout a number of sites in Southern California. And we developed a number of qPCR probes for detecting some of their target taxa, including arroyo toad, the 3-cent stickleback, the crayfish, pond turtle, And with these probes, we were able to identify the presence of some of their target organism DNA in a number of these sites all throughout Southern California. Okay, next slide. So as I mentioned, this was a small scale pilot study and it is ongoing, but it served as a really nice example of the benefits of partnering with water board staff or other environmental managers or stakeholders at the genesis of a project. And that really provided a number of training opportunities and a real collaboration in the study design. It also served to highlight the importance of proper training and consistent SOPs that were being used across a number of these different sampling efforts. And also it's going to help inform a better understanding of how eDNA is propagating in these systems, and that's help us interpret our eDNA signals going into the future. All right, so next slide, please. So there are a couple of different efforts that we have ongoing as well right now that are helping to aid in how we're interpreting our DNA signals to make sure that we're all doing it in a consistent manner. So right now we're involved in a number of different intercalibration exercises with both US and international partners that are focused on better understanding the influence of bioinformatic pipelines and also DNA extraction on the interpretation of our DNA signals. So for both of these studies, for the bioinformatic pipeline intercalibration exercises, right now we have a single DNA data set that's getting sent out to a bunch of different users that are all then analyzing it with their preferred bioinformatic pipeline. And then we take the taxonomic lists that are generated at the end of everyone's pipelines and we compare and look for the differences in how these different bioinformatic pipelines can eventually influence our taxonomic lists, which then eventually influences some of our bioassessment outcomes. Similarly, for the DNA extraction ring test, as they call it, we have some DNA samples that are have been homogenized and they all get sent out to a number of different labs, and we all extract that DNA with the same exact DNA extraction kit, but then we compare our sequencing results to see what organisms were present in each of the sample, and again identify where any of the different tricks or trades in everybody's lab protocols might be eventually biasing our taxonomic results. So both of these intercalibration exercises are helping us to identify what some of the best practice methods look like and how consistently they're being applied in a number of different user hands. All right, so I'll move on to the third area that I wanted to focus on, which is how we go about communicating some of our DNA results. So there are a couple of areas that I think we can really put our attention to, again, begin to bridge that gap between research and application. And I think this can be one of the ones that's the most intimidating, because as I mentioned before, when you get your DNA data sets, they can be large, they can be unwieldy. So how do we go about turning that into useful data that can be used to help inform management decisions? So the areas that I would focus on are on data management, data visualization, and as I mentioned, turning it into actionable information. Right here, I have a slide that includes a picture of a recent study that was published where the researchers did a large literature review to look at publications of environmental DNA-based studies, and they wanted to see what sorts of metadata people were including in the DNA that DNA results that they were reporting. So metadata really provides the context for where a sample was collected, how it was treated, how it was extracted, and all of that information is really critical for helping us interpret what the DNA results mean, but also potentially reproducing the analyses that those authors had performed. What this study found was that there's a huge chasm, I would say, or gap and differences in how everyone's reporting their metadata results. And right now there is not a consistent way in which folks are including that critical metadata alongside their DNA results when they get published. There are a couple of different efforts right now. I've included some of their pictures here that are focused on trying to standardize these different ways of metadata reporting. This includes something called the MICE guidelines for qPCR data and the mixed guidelines for reporting metabarcode data. Right now, we're really missing standardized metadata for eDNA samples. That's something that we're working on as part of the California Molecular Methods Workgroup. So really, for the novice user, this can be very overwhelming, but we're focusing right now on trying to make it standardized, at least for California, if not beyond, as part of some of these international efforts. And I really think it'll help ensure that the DNA data that's getting generated is going to be reproducible. Okay, next slide. All right, one of the other ways that I think we can help make users feel empowered to interpret their DNA data and get out of it what they need for discussing management actions is in data visualization. And so we'll be hearing a little bit more later today from Macy Lynn about the work of the Cal eDNA group, but they've been giving us some really great examples of what data visualization might look like. So for example, if you go to the Cal eDNA website, you can search for some of your favorite organisms that have been identified at a variety of different environmental sampling locations throughout California. You can zoom in on those individual sites where they've been identified, get a breakdown of what bacteria or other organisms of interest were also found at those sites. And you can also download a suite of the metadata that's included with the environmental sample. So this is really, kind of the gold standard of what we want to see for how we can be empowering users to play with their data and really be able to feel like they are able to interpret it for what they need. Okay, next slide. Another way that we can be turning this DNA data into actionable information, I'll give you an example here of another data visualization technique. This comes out of the QIIME 2 pipeline that you can use for interpreting your meta barcode data. And this again is another example where you're taking a data set that's sometimes terabytes in size and you're making it digestible for the user. They can play around with this, interpret their data at their own speed, and hopefully really feel like they're able to take this information and digest it and turn it into actionable information that can help inform their management decisions. All right, next slide. So for today, I think you're going to be hearing a lot from the different speakers in this session about how DNA methods really are primed and ready for integration into monitoring programs. And if you hit your next slide, and that we really can begin to close that gap between the researchers and application if we focus on a couple of key things like standardized protocols, joint pilot studies where we're partnering between researchers and the management community, and then also enhancing data interpretation and communication tools. Last slide. So with that, I would say thank you for attending this session. I, for one, am super excited to hear from all the other speakers in the session. And of course, as always, if you're interested in staying up to date on DNA-based work in California, you can feel free to email me to participate in the Molecular Methods Workgroup. So with that, I will hand it back over to Allie. And I can take questions or we can move right on to our next speaker. Awesome. Okay, great. I think that's keeping us right on time. And I'm going to go ahead and introduce Ann Holmes. All right. Great. So Ann Holmes is a PhD candidate at UC Davis. She works on aquatic organisms and trophic interactions using genetic methods. Who doesn't love that? She is interested in using genetics to inform conservation and management. Me too. And she's going to be telling us about enclosure experiments to inform eDNA interpretations in the San Francisco Estuary. All right, welcome, Ann.
All right, good morning. Thank you very much for the introduction. I'm happy to be here. And I'm excited to talk about some of my dissertation work about environmental DNA in the San Francisco Estuary. And as many people are familiar, the fish up here is a Delta smelt. It's a endangered endemic in the San Francisco Estuary and is of great management interest. So this work was done by our collaborator, me and our collaborators at the Department of Water Resources and the California Department of Fish and Wildlife, but we're actually piggybacking on an even larger study. So I'll thank those folks at the end, but the larger study was really what allowed us to install these enclosures and to bring in some cultured Delta smelt into the estuary. So let's see, maybe a bit of a lag here. Give it a second to advance to my next slide. Well, I'll start talking about my next slide. If I can, let's see. It doesn't seem to be advancing. Let's see, okay, there we go, thank you. As many of you know, eDNA is a sensitive and non-invasive method. It doesn't require morphological identification. There's a lot of advantages there. But it is more difficult to interpret when you don't have an actual fish in the net. And as Susie mentioned, we're interested in what does a positive detection mean, especially when we're in an estuary where we have a lot of flow, potentially a lot of turbidity, boat traffic, and things like that. So this question of how do the environmental conditions and biological conditions affect environmental DNA detection has been explored in a number of studies, but is underexplored in estuaries. And so that's what we're looking to do here, is have a controlled study where we know that there are some fish in enclosures, and then look at not only what the main direction of flow does, but the tidal flow and how detection is affected over time. Next slide, please. Let's see. So, so this is the San Francisco Estuary, and this boxed area is the study sites. There were 2 sites that we studied, 2 sites that we used, and this area is about more or less 100 kilometers inland from the coast. So the 2 sites I'll be talking about are both essentially on the Sacramento River. They're freshwater tidal areas, one near the city of Rio Vista and one in an artificial shipping channel. Next slide, please. And some of the questions that we were interested in investigating, mentioned before, should pop up in a second here. Can you hit the next slide if you're able? Let's see. Oh, okay, thank you. So, so as I mentioned, we're looking at how distance affects detection, so sampling at different distances away from the enclosures, as well as tidal action. So we're sampling not just downstream, but also upstream and in different tidal conditions. And we're sampling not only before, so we sampled before the enclosures were put in to make sure that there were no target fish in the area, but also after enclosures were removed to look at whether eDNA was retained in the area. Next slide, please. And this shows a picture of the 2 sites. And just very briefly, the setup was 6 enclosures, about 1 meter in diameter. They had about 60 fish each. And we collected 3 replicates of 1 liter samples at these different distances that varied from 1 meter to 100 meters, both upstream and downstream. As I mentioned, we're sampling before the enclosures were installed. And our detection was a single species qPCR. So just targeting delta smelt. We also did a lot of negative control sampling because we didn't want some unexpected contamination to influence our findings. Next slide, please. And so this is a setup of how I'm going to present the results. This bar here shows a hypothetical perfect detection if 100% of the replicates at each site showed a detection of delta smelt. And it's sort of a schematic where we have these different distances away from the enclosure. And this is showing a downstream sampling, it's downstream samples. Next slide, please. So what we actually saw was very strong detection close to the enclosure. So at 1 meter, not quite 100%, but 80-something% of our samples showed a detection of delta smelt, and it fell off pretty quickly. This isn't unexpected, perhaps, and at the further distances, we didn't have any detections. Next slide, please. And so the next slide, this is showing the same sampling date, but this is upstream. Just forward 1 slide. Great, thank you. And this is also perhaps not unexpected. Quite close to the enclosures, there was not a strong signal, but there was some signal upstream of the enclosures. So perhaps there was eDNA that had been left by a previous tide, or there's some different flow patterns. So even though the water is actually moving away from these sites and over the fish, there was some eDNA detection. Next slide, please. And so this is just a summary of what you've seen. We have these multiple sites above and below the enclosure. And what's going to pop up next is a different day of sampling, next slide, that shows what we saw on a different tide. So when the tide is moving in the other direction, despite the fact that net flow is obviously still moving out to the ocean, we saw something similar. And these are just single day samples rather than a summary, but this is a good indication that tidal direction does influence our detection. And next slide, please. So there was a pretty clear picture that distance mattered and tide mattered. And so this seemed pretty intuitive. And then when we removed the enclosures, we actually saw something quite different at these two sites. Next slide, please. So this was a little bit surprising. At one of the sites, there was a lot of detections, and at the other site, there were almost no detections. And so we went from having this very clear picture to something that was a little bit more confusing. These two sites are quite different in terms of the width, in terms of how the channel is used, and in terms of boat traffic. So, so that might explain the differing results, but I think it really points out the importance of habitat and how that might influence environmental DNA detection. Next slide, please. So just as to sum this up, distance was important. There was sporadic detection up to 100 meters, and that's not unexpected. There's other studies that show in other systems that eDNA can be detected quite far away. As I mentioned, tidal direction was important. And retention, I think, is something that probably requires some more inquiry, especially hydrological differences in these two sites. Next slide, please. And so just final thoughts, the distance and the timing that we saw sort of confirm that environmental DNA is a quite sensitive method, but that estuaries are challenging for environmental DNA detection. And this is what I've seen in field detections of delta smelt, and that's what led us to these more controlled scenarios. And based on what we saw, the presence absence seems to be a good, a good interpretation of detection rather than trying to quantify abundance based on all of these different factors that we saw that can change detections in a relatively small area. And as I mentioned, habitat variability is important. So, so basically, the positive detections were important, but I think the negative detections are important too. It shows that you can be quite close to a large number of fish, but depending on how the tide is moving, you may not see a detection. And I think this provides a lot of information, particularly for estuaries where you have this bidirectional tidal flow. And with that, next slide, please. I just want to thank the funding agencies, EWR and CDFW, and the many people who contributed to this study. If you're interested in learning about this study, feel free to contact me on the Slack. And I've also posted my my website there for some other work that I've been doing. So thank you very much.
That was perfect. Thanks, Anne. I think those are exactly the types of studies that are helping us interpret these eDNA signals. So thank you. All right, next up we have Kristen Ahrens, who is a research scientist with the California Department of Fish and Wildlife. She has a background in population genetics and biological consulting and is currently a part-time lecturer in the biology department at Sacramento State. Her current work is focused on the use of environmental DNA for fisheries monitoring and management, as well as population genetics of golden trout. I can't wait to hear all about CDFW's first in-house eDNA project. So with that, please take it away, Kristen.
All right, thank you so much, Susie, for the introduction. And I'm going to jump right into it because I have two projects that I want to talk to you about today. I'm going to start with the most recently and first completed project within our genetics research lab at CDFW. And I'm going to close out by touching on a grant that was just funded last month for a pilot study using eDNA. Next slide, please. So I'm going to start by giving you a little bit of background on the study area before I jump into the eDNA aspect. So the map you see here depicts the native range of a federally threatened trout, the Paiute cutthroat trout, or PCT. And this is a pretty small headwater area in the headwaters of the East Fort Carson River in Alpine County, California. This is only about a little under 18 kilometers in length, and this includes Silver King Creek and its tributaries. Next, please. And due to a long history of non-native trout stocking in this watershed, as is the case with many native fish in California, Paiute cutthroat trout were really impacted by the effects of these non-native fish that ultimately nearly led to their extirpation through predation, competition, and then in the case of non-native rainbow trout that were stocked there, we also had issues of hybridization. Next, please. So to remove these non-native and hybridized fish, much of this watershed had been treated with a commonly used fish toxicant, rotenone, over a series of years. Most recently, these rotenone treatments occurred from 2013 to 2015. And this was possible due to refuge populations that were free from non-native trout stocking. And that's what you see in the yellow lines within the watershed, these refuge populations. And ultimately, reintroduction of Paiute cutthroat trout throughout their native range was planned for 2019 within those rotenone treated areas. Next slide, please. So given this long history of non-native trout stocking, we wanted to use eDNA to address whether brown trout and rainbow trout had been successfully removed from the system. These results that I'm presenting are actually the third year of eDNA analyses. The first 2 years were conducted by the US Forest Service lab in Montana that helped us before we had our lab up and running. And efficacy of these rotenone treatments does vary, and it's best practice to confirm removal of the target fish. So this is where eDNA came in. And some of these areas within the watershed are notoriously challenging to electrofish, and they're high gradient, there's high turbidity. So this was an additional measure to really confirm non-native trout removal in a situation where the stakes are pretty high. Even if there's a few non-native trout still present, this could really compromise the costly and time-consuming reintroduction efforts that were planned. Next, please. So in the field, just to give you an idea of the scope of this study, we had 183 sampling sites across these rotenone-treated areas that were spaced every 100 meters. This was kind of based on the distance that some studies have shown eDNA travels, and it was on the very conservative end of this to make sure there were no spots untouched where we might be missing a single fish that could still be present in the system. And again, this is the third year of analyses across these exact same sites where these sites were sampled year after year. Next, please. Using 5 liters of water that was filtered in the field. So we took these filters back to the lab, and that is what we extracted the DNA from. Oops. So we used 3 target species assays ultimately. I'm going to focus on the brown trout and rainbow trout aspect, but we did also test for cutthroat trout DNA as well. So this was the qPCR approach. And we were just kind of wondering if we might have actually seen that some of the refuge populations were transported over barriers during high flow conditions. But the main focus was, again, these non-native trout. And so we use this quantitative PCR approach. We always use a series of replicates in these types of experiments because we're dealing with such trace DNA potentially. And we considered 2 to 3 out of 3 replicates to indicate that the target species was present. If there was only 1 positive detection, then we reran this sample 6 additional times. So to give you an overview of these results, in 2018, I did detect a single detection of rainbow trout across a series of ultimately 9 replicates, but we could not replicate this result. So while this made managers a little bit nervous, ultimately we considered this extremely weak evidence for presence of the target species. Again, we did this sampling over 3 years to give even more confidence in whether or not these species were present. And there was a bit of concern because in 2016, there was one rainbow trout detection considered positive at the downstream-most site. So ultimately, managers wanted to know, what do we do with this single detection? Should we be concerned? Again, we want to move forward with this reintroduction process. So ultimately, I would just emphasize that this eDNA data set should never be considered in isolation. We had 3 years of field data also to compare. We had electrofishing data in portions of the watershed. We had snorkel surveys, and they all also indicated no native trout presence. So what does this single detection mean then? This is what ultimately managers want to know. So there's always this issue of contamination that must be considered in these types of studies because we are dealing with such trace DNA. We have really stringent protocols both in the field and in the lab to prevent this. And we use negative controls to also help us to feel more confident that there isn't any contamination. And we didn't see any evidence of this. But there's always an additional issue that trace DNA could be transported from outside of the system. Downstream from the sampling area, we actually have non-native trout that are still being actively stocked. So could simply a mammal traveling through Silver King Creek be transporting some DNA on its fur? Could avian predators be bringing portions of fish into the study area? There's actually been a study that showed that fish DNA could persist for up to a month in avian feces. So these are things that we really can't control for, but we must consider. Ultimately, with these results alongside the field data, this really supported the reintroduction process, which began last year in September and is continuing over the next few years. By testing for cutthroat trout DNA, we also found that some of these fish, Paiute cutthroat trout, were actually present within the rotenone treated areas, likely transported over barriers during high flow conditions. So we're actually augmenting existing populations through reintroduction efforts. And eDNA is going to be used in a more targeted approach moving forward as this process of reintroduction continues. All right, now I'm going to shift gears and just briefly touch on this recently funded state wildlife grant that our lab received. That has two primary goals: one, to assess the native fish assemblage in the Lahontan Basin, and then to compare eDNA with electrofishing data. So the first objective is actually to develop an eDNA qPCR assay for mountain whitefish, and then to test some other special status fish within this native fish assemblage, including Lahontan cutthroat trout, mountain sucker, and Paiute sculpin. But ultimately, one of the major questions of this pilot study is going to be, how does electrofishing data compare to eDNA data collected on the same day? So we're going to select a sampling site within each watershed within the California side of the Lahontan Basin. So we have the Carson, the Truckee, and the Walker River watersheds. And it's really important for me to select some sites that differ in their hydrological and water quality characteristics, because we know that this can affect eDNA detection rates. And perhaps this pilot study will give us some information that tells us when it is best to use eDNA and when eDNA detections might be a little more challenging, or when they may be superior to electrofishing data. So ultimately, we're lacking a lot of distribution and abundance data on fish in this native fish assemblage, particularly mountain whitefish and mountain sucker. So my hope is through this pilot study, we can decide if we can use eDNA on a much greater scale and use this for a range-wide study of these fish, and that this could be our first step in addressing the insufficient data that we have on these fish. All right, thank you very much.
Okay, that was perfect. Thanks so much, Kristen. All right, I'm gonna move us on to our next speaker. We have Macy Lin, who is a PhD student in the Wayne Lab at UCLA, where she is also the host for eDNA Tea and Cookies, a weekly seminar series on eDNA research. Macy uses non-invasive genetic tools for conservation biology and has been doing great work as part of the Cal eDNA team. I am looking forward to hearing more about all this work. Please take it away, Macy.
Hello, I think I'm trying to start my video. Okay. Hi, sorry about the glitch earlier.
Perfect.
Cool. So I will be presenting the results from the Cal eDNA project initiated in 2017. I will be moving to a different perspective, mostly focusing on eDNA's potential for biodiversity mapping in a mostly terrestrial ecosystem. So to give you a little background on these projects, we all know California is a biodiversity hotspot, but still there is some lack of understanding of biodiversity across the tree of life. So our approach took advantages of environmental DNA and citizen scientists to actually achieve a relatively larger phylogenetic scope and larger sampling effort. Since 2017, we collected over 3,000 soil or sediment samples, and the data set that I will be presenting today includes around 300 samples collected during spring 2017. Cool. So our objective are threefold. First, we want to understand what was found in the eDNA samples that we received from the volunteers. Secondly, we want to understand how does the abiotic environment relate to the biodiversity patterns. We actually explored from alpha diversity to joint species modelings, but today, because of the time limit, I will be mostly focusing on the beta diversity patterns for this talk. Third, as every ecologist do, we want to fill in the gaps of the sites that we did not survey. We want to try to find the species in the sites that we did not found. But as we have heard from the previous presenters, that's a really challenging work. So we moved to try to predict community turnover from the eDNA patterns. So these are our three objectives. To achieve these three objectives, we first have to generate environmental DNA results with a meta barcoding approach. So as we receive soil or sediment samples from volunteers on their field trips, we extract the DNA from it, amplify them using 5 sets of primer that targets across the tree of life. So essentially, we did not only target like prokaryotics, we are also targeting groups like fungi and plants. We sequenced them on a Illumina MiSeq machine and generated taxonomic entries, which you could consider as a species equivalent using a software that we developed called Anacapa. To actually get an understanding of the environments of the sites that we surveyed, we compiled several environmental data layers that were known to either affect in soil microbiota studies or species occurrence studies. For example, we collected variables like bioclim, that are temperature and precipitations, and soil property layers, also vegetation indices as well. Here's what we found. To answer the first question of what is the diversity patterns, you can see here is a plot of the number of unique taxonomic entries found in each of the metabarcodes that we used, range from their target groups from prokaryotic to plants. Taxonomic entries could be considered as species in these talk, but there's some minor differences. As you can see here, this prokaryotic primer found the most number of species, and the proteobacteria and ascomycota are the 2 groups that are most commonly found in our data sets. To understand the diversity pattern and the abiotic environment, we actually explored several layers. Here, I'll be focusing on the variation in species composition among sites, which is we also call it beta diversity. We found variation in community structure in major habitat, as shown on this plot on the right. However, you can still see significant overlapping of sites. So we decided to zoom into each of these major habitat and try to plot the minor habitat scales. Now you can see there are finer scale partitions in minor habitats. Here I'm showing a zoom-in plot of the shrub-dominated habitats, and the chaparral and the scrub environment are separating on the first axis. We also explored the correlation with environmental measures as well. Here we are overlaying photosynthetic activities as the red lines here onto the fungal ordinations. And as you can see here, the photosynthetic activity actually separates the first axis of the fungal group's ordination very well. We also want to take advantage of the full biotic matrix that we have collected. So we used a different method called gradient forest. It's not actually a forest, but a random forest-based method. So it's able to take advantage of handling large data sets and correlated environmental variables all together, and identifies the community response and with the environmental gradients. So instead of the similarity matrix that we used in the traditional beta diversity analysis, we further cleaned the biotic occurrence data sets and converted them to species presence and absence patterns. The environmental measures were extracted from the 33 rasters that I showed before. What we found from the gradient forest analysis is that the gradient forest model explained a total of 35% of variations in the biological matrices. And the top three predictors are elevation, sand percentage, and photosynthetic activities. To answer the third objective of trying to predict the community turnover patterns, we used the abiotic and biotic relationship inferred from the last slides. We're able to transform the unstructured environmental variable matrices into according to the cumulative importance variables and turn it into a biologically informed community turnover matrix with a wall-to-wall coverage in California. We also colored the sites by the principal components. So on the sites, on the plot that I'm showing here, the sites that have closer color will tend to have a closer community composition. Here, sites A and sites B are more likely to have more closer community composition compared to site C. And here is what we found from the gradient forest map. So on the left is the gradient forest map. On the right are the EPA map and the USDA ecoregion maps. You can see that our gradient forest map were able to recapitulate a lot of the patterns from the EPA maps, such as like the Northern California coast, the Central Valley, the two deserts. So the main takeaway of the study is that we were able to explore different layers of the diversity patterns and found that environmental DNA is a good measure for biodiversity assessments. We were able to peek into predicting community turnover from eDNA by recovering a high resolution community turnover map like the existing ecoregions. I have to say there are still some limitations in the eDNA studies. For example, we also found stochasticity in the eDNA sampling as well. We found so a sufficient level of replications and incorporating variations into the models are very important as well. The 300 sites that we have included here is a good amount of sites, but still there's limitation of the sample size. So we are planning to reanalyze the data with the next stage of CalEDNA efforts. We are also trying to think about ways to ground truth the turnover maps that we achieved and try to actually apply the eDNA informed map for conservation management as well. Since then, CalEDNA has actually expanded a lot. So the projects that I want to mention here is the Protecting Our Outlet River. It's in collaborations of dozens of local NGOs and agencies. We were able to help with restoration efforts as well. Dr. Robert Wayne and Dr. Beth Shapiro has also developed an active learning class that used environmental DNA to introduce freshmen to career for conservation biology as well. My colleague, Mara Palacios, is also presenting a poster on the Mojave Desert Springs results as well in these talks. So it has expanded a lot. With that, I would like to thanks for my co-authors for their collaborations and guidance. Thanks for my funding agencies. And if you're interested in this study, you can check out our our preprint on the bioarchive as well. Cool.
Awesome. Thank you so much. That was great. Cal eDNA is really giving us so much great data across the state. So I look forward to all these future insights. All right, so that brings us to our last talk for today, and that is going to be a tag team effort from Joseph Crane and Peter Hoop. And so Joseph Crane is the co-owner of Jonah Ventures, which was founded in 2014 and helps clients answer ecological questions with DNA sequencing. And Peter has been with SWAMP for 3 years and is a recent graduate of the Oregon State University College of Forestry. So they will be tag teaming this presentation on using Jonah Ventures for answering bioassessment research questions. Take it away.
Good morning. I'm Peter Haupt from the SWAMP IQ unit. Next slide. To quickly review the SWAMP mission, we evaluate the physical, chemical, and biological quality of water to support a wide variety of users. Through carefully designed programs, we provide timely, high-quality data to assist other entities throughout the state. Next slide. In a 2020 program review, it was found that SWAMP's monitoring needs exceed their existing resources, and coordination between programs needs to be better integrated. This is where we think eDNA can provide great value to the program. Next. Metabarcoding can be implemented in a variety of ways and can drastically reduce collection costs, increase turnaround times, increase data quality, fill in data gaps like fish and other indicators, and improve coordination among programs using a single data type. Next. Metabarcoding gives us an opportunity to expand the scope of existing programs and provide a holistic panoramic view that incorporates a wide collection of species across the landscape, allowing us to examine the detail of a single species occurrence within a watershed as well as a greater assemblage at a single site. Next. And integrating soil, sediments, and biofilms can provide an even greater understanding of the ecosystem. Next. Commercially available sampling kits like those developed by Jonah Ventures, which you'll hear about in just a minute, provide simple standardized collection that can easily be implemented by volunteers involving the public in data collection in ways that could be complementary to established programs. These commercial services also provide accessible methods of data exploration as well as detailed spreadsheets for scientific analysis. Next. We're encouraging programs and regions to explore the benefits of eDNA and submit proposals to SWAMP. Next. Next. SWAMP will continue to contribute to the molecular methods work group, developing protocols and standardizing data integration. We also hope to expand our partnerships and contribute to larger efforts like the U.S. Forest Service EDN Atlas pictured here on the slide. You can see we're a bit behind our neighbors to the north and east. To this end, we've created a curated library, including journals and studies concerning field protocols, engaging the public, study design, and monitoring. This is available in a link I will post on the Slack channel. Next. The cost, efficiency, and comparability of eDNA data collection make it an ideal way to better coordinate between programs and allow our existing resources to meet our monitoring needs. Next. While the eDNA team was conducting research this spring, we were fortunate to be contacted by Nicole and Joe from Jonah Ventures and have been participating in their pilot project. Shuka and I have been using their sampling kits to sample from reference stations with historic data, and we'll be analyzing the results as they come in, but COVID and fires have slowed progress significantly. The kits are really easy to use and contain a preservative so that you can mail them in without any ice. This slide shows some of the Jonah Venture online tools with some of our sampling results, and you can see them at jonahdna.com. Here to tell you more about the project is Joseph Crane from Jonah Ventures.
Thanks, Peter. That was amazing that you got through that as quickly as you did. I had my doubts. Thanks, everybody, for listening for a couple minutes here. Just going to make sure that I can control screens. Potentially. All right, so just as a company, we've been around for 5 years or so. We process about 20,000 samples a year, and we've had over 350 clients that we've worked with. We'll do quantitative PCR as well as next generation sequencing, and we've got some new point of use tests that give you results back in 30 minutes that we're developing now. I'll talk about the next gen sequencing today. Essentially, probably the most important thing that we did is that we put everything in a box. So as far as all the sampling equipment is concerned, it's a syringe, there's a syringe filter, preservative, caps, gloves, mail-in envelope. We can get all that into a box. Anybody can use it. That's probably been the most important thing that we've done, even though it seems like the simplest. From there, we've paired that with smartphone apps on iOS and Android so people can easily record their metadata, real basic metadata, which can be expanded at a later point. It's all going onto the cloud into a non-relational database that can hold a lot of different data. And then that's really all an individual has to do is be able to suck up water, push it through a filter, mail it in, record some metadata. Then we'll do all the hard stuff as far as extraction of DNA is concerned, amplification, and sequencing, bioinformatics. Really briefly, we've had samples come in from all over the US. We're in almost all 50 states right now. This is from a couple weeks ago. But once you get your data back, all of that is accessible through the data portal, as well as we'll send out files to people too. So we've got some experience in a lot of different environments, not just California. And the real simplified version of what we present for fish is going to be a list of presence of fish species. We've got more detailed data that we send individuals, but on the app, or excuse me, on the data portal, you'll see that. And then we'll include images of the fish, its distribution as far as what's known, and then some descriptions for citizen scientists. It's helpful for them to learn about what they're finding. The success of this is I've been pleased with on the fish side of things, as well as phytoplankton. Just looking at the fish data, this is from a single site. These are the presence of roughly 25 species of fish that we found in South Dakota, just from 100 mils of water. It works pretty well. How many replicates we need, for example, to get everything that you would find is still open to exploration, but we're able to get a pretty high diversity from just 100 mils of water pretty regularly. And what we're finding in different places matches up with what are known to be in different places too. So this would be a South Dakota site. And then if we put it together for Alaska, for example, we're finding Alaskan species. It's pretty consistent. So I think we've got this platform relatively well worked out. These are just the fish data here. We're doing it for phytoplankton relatively regularly with just as high a success rate, and are working on other primer sets to look at different organisms also. Where we feel like this will have a rapid impact and a pretty major impact is, just like Peter said, is through bioassessment, and that's what we've really been concentrating on. Whereas with traditional techniques of collection, sorting for macroinvertebrates, for example, painstaking identification, generation of IBIs, it's expensive, it's slow, it doesn't give us good resolution taxonomically as well as for sites. But once we start to use eDNA, whether that's sucking up water or taking macroinvertebrates and putting them in a blender and just sequencing it, it's less expensive, it's a lot faster, and it seems like we're getting much more accurate water quality indices. So we think that bioassessment is going to move to eDNA at some point, and we're hoping to speed that up as fast as possible. And even right now, just with the fish data that we have on presence absence, we can use the data that we have for coefficients that represent water quality and show differences in water quality between streams that we find in New Hampshire, Alaska, and Oklahoma, just based on scores that other people have assembled for those fish species. Getting to IBIs and more sophisticated IBIs with eDNA is something down the line, but we're getting pretty close. That was my last slide, so hopefully I finished under time, and that'll give us time for questions. Thank you, Peter, for introducing everything.
Okay, that was great. So it looks like we have about 10 minutes that we can open up as a bit of a Q&A session for anyone that was in this eDNA session. So everyone's welcome to enter their questions onto the Slack channel. And I would invite all the speakers in this session, you can unmute yourselves. And as we wait for questions to come in on the Slack, I had one question I was going to ask Joseph and Peter. Maybe there's a question for Joseph. Can you talk a little bit about how you're handling on that last slide that you were showing, calculating fish IBIs? How does it handle abundances or relative abundances if you're working with meta-barcode data and you're looking at calculating IBI scores? I think that's a question that a lot of folks would have.
Yeah, it's what we found empirically is that relative abundance for fish from this approach of meta-barcoding, it's probably not that reliable, not yet at least. And so we're just working off of presence absence. And you might be able to get relative abundance from having replicates, but from an individual sample, it's just a list of presence. For phytoplankton, relative abundance is a lot more possible because there's a lot more DNA in there. And so we feel like getting more accurate IBIs from phytoplankton is probably going to be, and again, we know that different organisms provide different signals, but it's probably going to be the future more than fish. But at the same time, we can get fish, macroinvertebrates, phytoplankton all in the same fell swoop. And so more than likely, those IBIs will work across all those organism groups. Thanks, Susie.
Awesome. Okay, we did have one question that just came in that asks about local eDNA libraries. So are those needed in small headwater streams? Do you need a certain number of individual species to get a good ID of species? I participated in a study at OSU. This is from Cajun James. We had trouble with low flow numbers of species. So let's take that question in two parts. First off, I'd love to hear from any of you all on what about local eDNA libraries and if they are needed. Do we have any strong feelings about DNA libraries? I know I do, but feel free to speak up.
If I can jump in,
Yeah.
because we've worked on this pretty extensively. Right now, it's not just local libraries, it's global libraries. There's somewhere on the order of 1,200 fish species in the US, and roughly a third of those have been sequenced. That's it. So we don't need necessarily local libraries, we just need the ones that have each species once. And the phytoplankton side, it's even worse than that. It's probably 5% of the species have been sequenced. And so we get pretty close, but even California with its reduced number of fish species, for example, that are there, there's still a whole bunch that haven't been sequenced yet. So yeah, we've got a ways to go just to get to the first pass. I don't know if other people have opinions on that.
Yeah, I can speak to that too. That's something that that my lab at UC Davis is involved in. And my PI, Andrea Schreier, received a Prop 1 grant to actually put together reference libraries for the San Francisco Estuary targeted at fish and larger macroinvertebrates. And we see that as a really important part of moving eDNA into a place where it can be more useful to management.
Yeah, another data set that I know of is developed by my colleague Zach Gold called FishCAR. So it's a marine fish 12S California specific reference database. So I think it's definitely like very important to construct local reference DNA thread libraries if you see that you have trouble finding the number of species, or you have trouble like finalizing, or for example, if you it's possible that you found match to like family level for like a different species at a different locality. That's like where it's necessary to have those.
Okay, awesome. I just got another question that came in that says, can you get terrestrial plants in aquatic eDNA? Is there anyone that wants to take that question?
Yes, you can. And one source we think is through pollen, but I think it's also possible to detect plants through detritus or just bits of material. But yeah, definitely.
Yeah, I think the idea of the autochthonous versus allochthonous input of eDNA is a big one, an active area of research. And a lot of folks are interested what can be getting washed into your watershed if you're looking at aquatic eDNA. So I think that's an active area of research. But the short answer is yes, you can get terrestrial DNA in your aquatic samples. Any other comments on that?
I'll just add to that we could take that as far as terrestrial animals or mammals as well, especially in areas where maybe there's not frequent water sources for those mammals, and we know that they probably are visiting a certain aquatic area often.
Awesome point. All right, so one of the other questions that I was hoping that we would hear from the panelists about are, what's the area that you would like to see the greatest amount of research efforts focused on in the next 5 to 10 years? So maybe we can just go through everyone. If you have strong feelings on where you think our eDNA field really needs to concentrate its research efforts right now to help advance eDNA and the integration into routine monitoring efforts. I can start off with my opinions. I think what we heard a lot today already was that some of the DNA reference libraries can really be constraining our ability to identify some key target taxa in our samples. I think to a certain extent that's some low-hanging fruit. We know we're good at sequencing reference sequences. It's just a matter of devoting the time, effort, energy, dollars, and hopefully being able to actually sample those organisms that we need those reference sequences for. So I would love to see some really big progress in that arena so that it's just something we don't have to worry about in 5 years. Anyone else?
Yeah, I'll add that I'd really like to see more data on using eDNA for abundance estimates, which we're already seeing a lot of publications on this, but I think in the next 5 to 10 years, we're going to really refine our ability to use eDNA to create these abundance estimates. And then also looking at doing like RNA-seq or getting at gene expression ultimately from a water sample potentially, and how that might have implications for ecosystem condition and how gene expression might be changing, especially for thinking in California, post-drought, post-wildfire, things like that.
That would be so great. That would be such an exciting area to see some more development. We talk a lot about it in the harmful algae world. If we can get at RNA transcription as an early warning indicator, so that would be awesome. Anyone else?
I think your study on fate and transport is really important, that, you know, we can sort of determine how far this is moving. I thought the studies today were great.
Awesome. Yeah, exactly, Peter. I think that's something a lot of folks would love to see. And in the increasingly more complicated arenas also, like estuaries and beyond, where we're dealing with our tidal regimes, that would be awesome. Anne or Meiji?
I would think that SOPs is the most important thing, like next steps. And maybe also trying to, like, we do a lot of experiments to test like the transportation distances, to test like the replicates and the stochasticity in them. I think it'd be important if we could model them to some extent so that we don't have to completely use empirical data to inform our choices.
Awesome.
Some things that I see are on the communication front. So communicating not only the potential, but also the limitations. eDNA is, you know, it's another approach, and it, like all methods, it has some limitations. And I think that will help integrate eDNA with other methods where it can be complementary. Yeah.
Awesome. Awesome. All right, I see another question that came in that is from Keith, and he asks, have people gotten eDNA detections at distances over 1,000 meters? What is the maximum spatial scale people think eDNA can be detected in aquatic habitats? Does anyone want to take that one?
Yes, I've seen those numbers published, and I think it's very possible. I think it's probably varies a lot on habitat and species. You know, how abundant was the species to start out with? How does the transport work? But yeah, there's a lot of really good questions to explore in those long distance transports.
Yeah, something that comes to mind thinking about long distance transport is a study done by Christy Diner that they looked at distance of travel of, I want to say an invertebrate species, maybe Daphnia, that was transported from like a lake outlet downstream up to 10 miles in this stream. So possibly really long distances, but for fish studies, it doesn't seem like we've shown that that we have that long of a distance necessarily.
Yeah, I think across the board, we're seeing there are so many different environmental parameters that go into dictating how far that eDNA can travel and how long it persists in the environment. So I'm really excited to see, hopefully in the next 5-10 years, our ability to constrain some of those estimates so that we have, some great models that we can plug some numbers into and get a good estimate for our target environments on how long we expect that DNA to exist. And that'll help us determining our sample schemes for years going forward. So with that, I think we're at time so that we have enough time for a break with everyone. But I would like to thank everyone that participated in this session. I think the talks were really interesting, and I think it gives me at least a lot of great ideas and where I see our eDNA work going in California. So it's great to have you all involved, and thank you so much.
Susie.
All right, thanks, Susie, and thanks, everyone. Let's come back here at 10:25. All right, good morning, everybody. We're going to go ahead and get started up again. This is Allie again, and I'm going to just quickly introduce John Olson. He's the moderator for our next panel, or it's not a panel, session of talks. So John's a freshwater scientist at California State University, Monterey Bay, and he studies the ecology of streams and rivers and how they're influenced by the landscape around them. John also examines freshwater ecosystems using a variety of tools like DNA, satellites, and models to better understand how they function. And then he then applies this knowledge to developing ways to improve the management and health of rivers and streams. And with that, John, I'm going to turn it over to you. And then you're going to turn it back to me.
Yeah, so Allie, my first task here is actually to introduce Allie. Allie is a senior environmental scientist of the California Water Boards and the Surface Water Ambient Monitoring Program Unit Chief. She attained her degree from Cal State University, Sacramento in biological conservation, has nearly 9 years experience working with natural resource conservation and watershed management for the state of California. And she's going to be talking to us today about the current early look at what's coming out of the SWAMP program assessment.
Cool, thank you, John. And thanks for that introduction. I will just briefly be talking about current efforts with the SWAMP bioassessment program, and then give a little sneak preview of the most recent results for the Perennial Streams Assessment Report, which was developed by Andy Wren and others. And Andy's also on call for Q&A if there's questions I can't address if and when we get there. So just very briefly, I think, you've all heard a lot about bioassessment today, but for those that are new to the program, what we're looking at is looking at water body condition based on the things living within that system. So primarily we're looking at macroinvertebrates and algae. The bioassessment program through SWAMP is many things, but there's two core monitoring programs, the Perennial Streams Assessment and the Reference Condition Program. And so with the PSA, that's random sites throughout the state looking at long-term trends, and then the reference program is more targeted monitoring, looking at water bodies that are in reference or good condition. And so, like I mentioned, we are primarily looking at wadable streams, mostly perennial, and the way that we're using that data, and I'll just speak for the state boards program, but the way that we use that data is a couple of ways. One of which it helps inform our biological objectives. So in past years, you've heard from the San Diego Regional Water Board. They've done some work in that way, but it's also really critical in integrating different data sets. So being able to use that biological data for these more holistic assessments of watershed condition too. So just a couple examples. If you're not familiar with the program, go poke around on our webpage there. And there's lots of material there that you can peruse at your leisure. Okay, so what we're up to currently, the kind of key efforts underway is really focused on improving the data lifecycle of bioassessment monitoring. So wait one second, I had a little, bear with me guys, I just had a weird crash. Wait for it. Okay, so we have done a pretty good job as far as getting out there and collecting and processing data, doing a lot of this analysis, but we're really focused on the preservation and storing this data in a centralized place, to making that data available publicly and in an open way, and then pushing that out there, publishing it, letting people kind of discover that data and integrate with other programs. Through that, then we're hoping to be able to develop more of these data visualizations. So down here at the bottom left of the screen is a prototype we have, just trying to get the data communicated in a way that makes sense to multiple audiences. So folks that maybe aren't familiar with biological data, we want to say it in a way that makes sense to them. Okay, so kind of along those lines then, we just recently, a team of us, updated the instructions for calculating those indices. The first draft of that primarily just focused on the CSCI, whereas this is pulling in the ASCII and the Index of Physical Integrity, or IPI. That draft will be available, we're hoping, by early November. And again, I just plopped the webpage there, so feel free to peruse that when you get some time. Second and last update then is the ASCII calculator was revised back over the summer. And that package is available on SWRCB's GitHub page, which I have listed here. And when I go over the PSA report, you'll see that I left ASCII out because of those revisions. We're recalculating the ASCII scores statewide. So stay tuned for the final report, which will have all those things there. Okay. So jumping into the PSA results, the PSA is really focused on these 4 primary questions. I won't read them, we'll go over the results through the next slides. And it covers this 11-year time period between 2008 and 2018. The design is, like I said, it's a probabilistic design with random sites throughout, and each survey site represents some portion of that total perennial stream length in the state, or its weight. The survey is stratified by region, land use, and stream order. And there are several different indicators that were used that went into that. We're not using ASCII, as I mentioned previously, but the CSCI, IPI, and CRAM I will touch on today. There are several data sets that were also used throughout the state, and I just wanted to emphasize here that we have the PSA and the PSA through the bioassessment program, but there's also several survey partners, and their data has also been used to compile this report and the assessment. So that's the key take home there. Okay, so jumping into some of the results. So this is really getting at the biological, physical, chemical condition of streams statewide. And so just, I'll try and keep this brief, but if you look at the CSCI, about almost 50% of the perennial wadeable stream length in California was likely intact for that 11-year time period from 2008 to 2018. If we look at IPI and CRAM just being another measure of the physical attributes in a stream, you kind of see that the IPI might be one of the least sensitive indices, in that it had more than 50% of the stream length likely intact. Whereas with the CRAM, you see a considerably different story. So there's only, just over 30% or so of statewide stream length that scored is likely intact, which is lower than the other guys. So this slide is just showing, if we break that down by each of the ecoregions, the CSCI, that percentage of stream length is in that best biological condition through 2008 through 2018 really varied across these different ecoregions. So just to highlight some of those, if we look at the North Coast, for example, the Sierra Nevada, those both being on the right-hand side of the screen, center and bottom, those have the highest percentage of likely intact. Whereas if you look at Central Valley, most of the stream length is not doing so hot. And that's a trend we've seen over time. So this bigger question then of, are stream conditions changing over time? So I organized these just by each of the indices. And for the CSCI, if we look across all years, you'll see that there's kind of the stable trend across time. Whereas with some of the regional results, the different regions really vary across the state. So there's, North Coast, again, Sierra Nevada, you see that they're still pretty likely intact over time. Whereas, you know, chaparral might be getting a little worse for that likely intact over time. And in the interest of time, I'm not going to go over all of these results. Okay. Okay, so for the IPI or the physical attributes then, we see again, similar trend there where things are relatively stable over time statewide for all years. The regionally, again, there's some variation. So as an example of that, if you look at the chaparral, this top left, or we look at the North Coast and the Sierra Nevada, things are staying pretty much the same in better conditions. And sorry for jumping over this, but there's a lot here and not very much time. For CRAM, again, another physical attribute measure that we have. The trends are pretty relatively the same over the time. Central Valley, you see there's really a lower amount of intact going on. North Coast, again, staying pretty good. Okay, and so this third question about, we're looking at the relative condition of streams statewide. Okay. So the sites just quickly were classified into these 4 different land uses: ag, urban, other, and forest. The main take home from here is we see a lot of the ag and urban dominant watersheds are really impacting biological conditions and the physical attributes, whereas in the forested watersheds, things are a little bit more intact. And that's just another slide saying those things. Okay, so this piece is really important, especially from a management perspective in my mind. So just to touch on this, we're looking at relative risk. And so this is essentially just on the bottom on the x-axis is showing those different stressors that are impacting biological condition and the risk associated with that. And the take home here is the physical attributes. So we look at the IPI, the riparian veg index, and in-stream habitat. Some of these physical attributes are all really driving biological conditions. And that's important from that management perspective, like myself, like, well, what does this all mean? What do these results mean for me as a manager in the program that we're overseeing? Okay, so just to quickly run through those recommendations then. And before I jump into this, looking at the physical attributes, I mean, that's important to know if as we're modifying these systems and we're seeing these impacts of biological condition, that has implications for how we're managing things through, at least at the water boards, through our stormwater permits and how we're changing the landscape. That could help us say like, hey, you know, maybe slow down on development and some of these things. Even though that we saw that over time, things aren't really getting worse, but they're not getting better, it's not necessarily saying nothing. And so the take home recommendation is we want to continue the statewide regular monitoring throughout time, but then we ramp up more at, you know, local level to see what's going on at those local conditions or local sites. So one thing we get asked a lot too is in regards to climate change and how biological conditions are changing. So one of the key recommendations from this report is instead of using the CSCI or ASCII, that we'd want to look at some of these raw metrics or at the taxa list to be able to say something about how sites are changing due to climate. So that's something that we're interested in pursuing. And then there was some discrepancy between the CRAM sites or the CRAM results and the other indices. And so one recommendation is let's take a closer look at CRAM and some of those sites and really weigh, you know, what are we getting from those results? And are they kind of making interpretation too challenging? Are they really saying something? We just need to take a closer look for management so we know what's going on. Okay, and with that, I think I'm at my time. So thank you, and I'll hand it back to you, John.
Okay, Ali. So there was a question by Kara Clark asking about the CRAM data for the desert's mudrock having a large drop in the proportion of likely intact streams, and wondering what you thought was going on there. Rafi's actually already chimed in a few times to answer that a little bit. Rafi was pointing out the fact that you know, very large sparsely sampled region, so one site can change and have an outsized effect on, you know, the overall regional pattern. So basically, you're seeing change driven by a single high-weighted site. Do you have any more to add to that, Ali?
I can try, and then Andy can jump in. But from what I understand too, there's a low number of sites there, and they carry heavy weight. So if there's 2 sites and they are, you know, not likely intact or likely altered, then we're going to see that. Andy, did I get that right? And if not, go for it.
No, that's absolutely right.
Got it right. Cool.
So, Ali, I was
Super
wondering.
nice presentation, by the way. Ali, I just want to say that was really nicely done. Thanks for covering that. You did a great job.
Thanks, Andy.
So, Ali, I had another question. You know, so we've been listening yesterday about, you know, intermittent and dry streams, and we've listened today about eDNA. You know, it's kind of interesting what your thoughts are. You know, what's the timeline do you think, or what would be the approach of beginning to integrate these different ideas into bioassessment within California?
Yeah, I mean, it's definitely, we all know California is a very intermittent, dry, ephemeral state. So it really does boil down, not to be the manager that says it boils down to resources, but we do have resource constraints. It's not that it isn't a priority, but that's partly why, like, if we do this assessment looking at, we have the IPI, ASCII, CSCI, and CRAM, reassessing the need for, you know, if we can get away with maybe not doing so much CRAM at every site, maybe those funds are released to do some of the ephemeral work. So it's definitely a priority for us. It's just figuring out how we get there. And I don't have an exact timeline, John. I wish I knew, but I don't know.
I was just curious. So a question from Rafi Mazer is, what are the different priorities for the reference program, both within the White Water Board and with other partners?
Again, yeah, so it's kind of similar answer. I know within the water boards, there's been some interest by some of our board members. Just climate change is important to all of us, but I think using those reference sites as a way to assess over time if things are getting better or worse, that would be really insightful and valuable. So I'm just speaking from water boards, it's again something we identify as a priority and would like to put more into, but yeah, resources.
Well, this next question might have the same answer again, but I'll ask it anyways. So how much, well, kind of what are your thoughts on, you know, when you look at different stressors happening within California stream systems, are there ideas about expanding the number of stressors to include, you know, new novel stressors like microplastics, or, you know, some of the new pesticides that are coming out, and trying to identify what these other type of stressors effects might be on stream systems?
Yeah, I mean, I think we'd like to do that, but right now the key priority is we do have this mountain of data and this really foundational data, just trying to get a grasp on our current focus with the program and looking at the stressors we do know about and biological conditions, and getting that data to be more regularly used within the water boards and others. So I think once we pass that hurdle and get over that bridge, then we can pull in more things, but trying to keep our focus so we don't try and do a million things sort of good.
Okay, right.
Yeah.
So one last question from Cajun James is, for the condition category is not known, is that due to lack of sampling or access, and what might be causing that problem?
Andy, you want to chime in on that one?
I'm sorry, can you repeat the question, John?
Yeah, if you ask questions about the category not known, is, you know, there's lots of not knowns within these data sets, the gray bar at the top. Was that due to access problems, or simply, you know, you couldn't sample it for some reason?
It's because not every survey collects all data types. So for example, the U.S. Forest Service Partner Program doesn't do CRAM. Certain programs haven't done algae over time. So, you know, for the certain years and certain programs, entire data sets are missing.
All right,
That's
thanks.
the biggest. And you do have the occasional case where, you know, like a sample jar is broken or whatever, and so you'll get like, you know, random missing data, but mostly it's because it's a programmatic thing.
Okay, thank you. All right, thank you, Allie. I think it's time to move on to our next talk.
All right, thanks, John, and thanks to Andy and Rafi.
Yeah, thank you, guys.
So Anna Holder is an environmental scientist with the Office of Information Management and Analysis of the State Water Resources Control Board. She's a program coordinator for the Surface Ambient Water Monitoring Program's Bioaccumulation Monitoring Program and the technical lead for the Healthy Watersheds Partnership. And is also a statistical modeler with the Desert Research Institute and has obtained her MS and her BS from Cal State Monterey Bay in applied marine and watershed science. Regardless of her role, Anna uses data science, ecology, and science communication to contribute to and inform sustainable management of natural resources. And she's going to be talking to us today about some work we've been doing in my lab looking at fish distributions across Alaska.
Awesome. Thanks, John. All right. Well, I'm just going to dive right in. But first, I'd like to acknowledge John and I have been working on this along with Arev and Jesse, and all of our funders and partners. As all of you know, I'm sure you can't really do research alone. So it takes a village, and this is part of our village. And so I just wanted to acknowledge their efforts up front in case I run out of time. Although I'm gonna stick on time. So this study was conducted in the National Petroleum Reserve in Alaska, which is on the remote North Slope, and shown on the screen here, and is over 22 million acres and contains thousands of miles of streams. The NPRA is the Bureau of Land Management is tasked with managing oil and gas leases in the NPRA. And this figure here shows the leasing results for 2019, and I don't expect anyone to read those itty bitty numbers in each of the boxes, but the take home here is that the rapid increase in development is shown in the past few years, is shown in the blue and red and like pink areas, and everything in white is potentially open for leasing in the future. So a lot has been happening recently, and a lot more could happen in the coming years. So the Bureau of Land Management is interested in understanding how fish species are distributed over the landscape. They're interested in the 19 species listed on the screen, but the top 8 in white, and also shown in pictures on the right-hand side, are of specific interest for ecological or economic reasons. And so the Bureau of Land Management, tasked us to develop species distribution models over the NPRA landscape to help inform their management decisions in the future. So a little bit about species distribution models. Generally speaking, they compare known species occurrence locations, usually in the form of presences and absences, with corresponding environmental conditions in the form of raster layers, and you mush those together, do some fancy math magic, and you get your potential prediction of species distribution over space and time. But in the NPRA and many other spaces, it's complicated. I'm sure you're aware, fish are in streams. So lotic environments include, all the upstream area. And so looking at the space in terms of a single point in time or a single grid doesn't really capture what the fish might actually be experiencing. And they're on the North Slope of Alaska, which is really difficult to access, making traditional survey methods costly. and resulting in a less than ideal amount of data. And, you know, they're not trees, they move, the little stinkers. So they're hard to detect through traditional methods as well. We can, I'm sure, debate about what an absence truly means in a method. We can talk about that later, but we attempted to overcome these challenges by altering the traditional approach to species distribution modeling by doing a couple things. First, we used environmental DNA, which we had a great session earlier today about. We used environmental DNA surveys in addition to traditional surveys to try and increase the efficiency of sampling and detection when in the field. And we used averages of satellite observations over longer and different timescales to increase the amount of usable data for the study area. And then we also use landscape characterizations from StreamCat, which converts the environmental conditions from those rasters that include a single point in time to linear stream segments that take into consideration the entire watershed, which I'll talk about in a moment. And we use maximum entropy modeling to develop species distribution models for all species of interest to produce species predictions over the NPRA landscape. And I'm going to walk you through each of these steps in a little bit more detail because there's a lot there, so hang tight. First, we'll talk about the satellite observations. As you're aware, a satellite gives you a snapshot of environmental conditions associated with a single point in time. And I'm going to talk about static and dynamic predictors here. And when I refer to static predictors, I mean those variables that were summarized over the entire time period that we looked at from 1979 to 2016. We had 96 predictors. And then the dynamic predictors were summarized on ecologically relevant timescales preceding the sample, which allowed us to account for both spatial and temporal variation within the study period. And then so that data was restricted to 2000 to 2016. And we used, you know, the time of fish occurrence, 2, 4, 6 weeks prior to the sample, and then 3 months preceding the survey. I also mentioned StreamCat. So once we collected the satellite imagery, we ran it through StreamCat, which summarizes landscape variables for each subcatchment that you can see in the figure here. And it summarizes the variables to include upstream catchments, so you can kind of get an idea of, you know, the upstream experience that might be impacting the actual sample location. And that results in watershed averages for each stream reach, which you can kind of see the little lines in the maps there. And that takes us from environmental data in the form of gridded rasters to environmental data in the form of linear segments. And then StreamCat, in combination with the temporally dynamic predictor data sets that we used, really could enable us to understand whether and to what extent there are lagged influences to predictions of species of interest. And more importantly, StreamCat allows us to calculate environmental conditions for all stream reaches in the study area, which is huge and really important for the MaxEnt method. So MaxEnt sometimes gets a bad rap for being a little bit of a black box method. So today I'm going to try and shine a little bit of light so that we can work through the technical darkness that is MaxEnt. So first, the principle has been used in information theory and statistics since 1975, when Edwin James suggested that we really should only use the information that we absolutely know in our models, meaning the presences. And to estimate the distributions or absence points, also referred to as pseudo absences, using the background environmental information, so that in our sense, all summarized streams produced in the StreamCat would be considered that background information, and we would use that as the area for pseudo absences, and then the actual presences as the presence locations in our species distribution modeling. The benefits of using Maxent include its ability to use presence-only data sets, as well as highly collinear data sets, and still produce pretty high-performing models. The critiques, there are many, but one includes its black box application, but a 2017 release made it a little bit more transparent and easier to kind of get inside there and figure out what is happening with your models. And there's also a tendency for overfitting, but that can really be addressed by your predictor selection, so what variables you're going to use in your model, and through tuning of something called the regularization parameter, which controls the complexity or parsimony of your model. So for our modeling process, I'm going to go through a conceptual diagram here. We started with an iterative predictor selection process that is based on the permutation importance and percent contribution of the variables, as well as their correlation values. We then looked at regularization parameter sensitivity to understand how much we should tune the models. And we evaluated the models using a 10-fold cross-validation, where we considered the number of performance indices, but gave specific weight to area under the curve and other, the number of predictors in the model, lots of other things. And we wanted to compare the Maxent performance in a couple of different ways, using the presence absence method, that's a little bit more traditional in species distribution modeling, as well as the presence background, which is a little bit newer with using Maxent and StreamCat, the static versus the dynamic data sets. And we compared all those things using the variables or the performance metrics that I have provided below, and we'll kind of go through some of those. So with modeling performance, the take-home message here, that we've got a couple of different metrics and the data set type on the x-axis. And the big take-home is that you can see the red arrows going up. That means that the models that use presence background data and static models were the best in all the different metrics. When we looked at the regularization parameter sensitivity, again, there's a red box here kind of highlighting the sweet spot where you have the high performing models that don't compromise model complexity. And so you get kind of the most bang for your mathematically numeric buck, I guess we could say. And that sweet spot was between a regularization parameter value of 1 and 2. 1 is the default, and 2 makes it a little bit less parsimonious. When it comes to the variables that showed up as being the most important, we have the core ones were temperature and land cover, and there's some details here regarding like how frequently they were used, the response in the model, and their permutation importance and percent contribution rank. Potential evapotranspiration estimates were the specific predictor that kind of shot up to the top in terms of permutation importance, but you'll notice it wasn't used that frequently. The most frequently used variable was used 13 times, and that was distance from the stream segment in the coastal foothills, which we can talk about what that means. The best part about all this is we can develop species distribution maps that I've shown here for 4 species. And so the cooler, bluer colors indicate a higher prediction of presence in that space. And it varies based on the species. And I think these maps in general can be and will be used by the BLM to help inform their decisions. So we found that Maxent is a very powerful tool, and it works well, especially when you use it as it was intended with background data. And we found that tuning the regularization parameter is useful, but not necessarily critical for many species. With the dynamic data, where we kind of cut the data up into different time periods, that was a lot of work, and it didn't really improve the models that much with its use. So it was kind of underwhelming. We're a little disappointed in that. And like I mentioned, the maps really could inform management decisions. And I know I've been, drowning on and on about all this. First, I'll say that if you are interested, you haven't gone to sleep yet, I definitely recommend checking out a paper that we recently published that kind of goes into the details of everything that I mentioned here. And the supplement has a lot of the maps and the analysis of the regularization tuning and all that good stuff. But this was in Alaska. And just like here, Alaska is being impacted by climate change. And so we're now looking into those impacts and seeing how the different species could be impacted by increase in temperature and other variables. And in California, I think the application could be really great. The methodology can really be applied anywhere. We talked about using eDNA and bioassessment data. You can potentially use Maxent to fill in or impute data gaps where samples are lacking, and conduct landscape scale assessments to figure out where the different species are. And, you can use it to also predict climate change impacts in the future. Thanks.
Actually, I think you might have answered Rafi's question, but let me give it to you anyway. So Rafi asked, what would be the best uses for Maxent modeling for bioassessment? You started getting at that at the end, but can you expand upon that a little bit?
Yeah, so I think we all kind of suffer from lots to do and not enough resources to do it in. And so I think Maxent could be really powerful in using the data and sampling methodologies that we have and kind of expanding or imputing missing data throughout the state. So the sampling programs are great and we should definitely have them, but there are large swaths of the state that don't have a lot of data, and we can kind of inform our management by filling in the predicted bioassessment, species locations and other places.
So Rafi followed up with, what is the most promising approach that came out of the work we did in terms of bioassessment?
Yeah, I think what was really promising was the combination of all of these methods. And so not just using Maxent modeling, but in combination with the landscape characterizations through StreamCat, being able to assess an entire landscape with robust methodology and good data to inform and produce really high performing predictions, which I know it's like a hand wavy, everything's great answer, but I really do believe that the combination of all of our methods is the most promising piece.
Okay. Steve Moreno has a quick question, or two quick questions. First, how does the approach handle interspecific interactions, such as between whitefish, cisco, pike, and salmon competing for niches or prey or on another? Yeah,
so we looked at, when we did the modeling, we looked at one species at a time. And so we're I think the next phase is digging more into those types of interactions and into richness and how the complexity of the environment, which is like real and exists, but you can only do so much in a model, right? So I think that's kind of the next step of really digging into what those interactions look like and how we can robustly account for them in the model.
All right, thanks, Anna. I think we'll have to skip the second question. He'll leave it on Slack for you to answer.
Awesome. I'll head right
Move
over.
on to Jeff Lauder. Jeff has a background in tree physiology, climate science, forest ecology, aquatic ecology, and statistical modeling with more than 10 years of research experience in the forests of the Sierra Nevada. His current interests include examining how climate change has already impacted and may continue to impact the Sierra Nevada ecosystems, ranging from water quality and stream health to forest drought and resilience. So we'll turn it over to Jeff.
Yeah, hi, can you hear me? Good?
Yep.
All right. So yeah, my name is Jeff Lauder. I'm an ecologist with Sierra Streams Institute. Thanks for having me this morning. I'm going to be sharing some of our work on climate change impacts on a Sierra Nevada foothill watershed, specifically using our 2 decades of citizen science data to kind of get at this question. So we are a citizen science group in the Sierra Nevada foothills in Nevada City, California, that's been monitoring the Deer Creek watershed using a team of entirely volunteer citizen scientists, monitoring 17 sites, most of which since the year 2000. The Deer Creek watershed is a perennial foothill stream located in western Nevada County, where at all of the sites shown on this map, we sample all of the parameters listed on the screen according to the Surface Water Ambient Monitoring Program protocols. Should note all of these are measured by trained volunteers using calibrated sensors and then QA/QC'd in-house, except for nitrate, phosphate, and bacterial concentrations, which are sampled by the volunteers and then brought back to the lab to be measured by us in-house as well. We then also do biannual benthic macroinvertebrate sampling, and we identify down to the family level, being a citizen science group, and we also have community members come in to assist with this identification and bug counts in-house as well. And then some additional analyses that I'm going to be sharing today involve some land cover analyses. So at the catchment scale, kind of actually following the stream cat flow that Anna was just describing, where any catchment scale data is at within that site's catchment and all upstream catchments. We looked at forest cover in 2001 through 2016, agricultural cover, grassland, all the variables listed on the right there, and then also extracted normalized differential vegetation index, which is just a proxy for canopy cover along a 30-meter buffer along our stream corridor. And so using all this data, we definitely as an organization like to ask a lot of the same questions that other watershed monitoring groups ask, just monitoring our stream over time, which sites are sensitive to change, which sites are resilient, and just keeping on any like point source pollutants and stressors. But obviously the big elephant in the room and question that we'd like to start getting at is related to climate change. So we do know that over the last 100 years, mean and minimum temperatures have been increasing, as shown on the top left here. And then this leads to a concurrent increase in evaporative demand, or actually evapotranspiration, shown on the top right. And then a variable that we're interested in is climatic water deficit, or a measure of aridity, CWD. So you'll hear me refer to that throughout the rest of the talk also, shown on the very top right there, that has increased significantly over the last 100 years in Southern California, coastal California, and the Sierra. And then further, we actually know that in our 2-decade data set, we've actually experienced some pretty severe, pretty extreme climatic events that might help us to bound expectations of what may happen in the future. So for example, 2012 through 16 was the one of the most severe droughts on record, the California mega drought, and tree constructions of Palmer Drought Severity Index on the bottom left there, with more negative being more dry, show that going back 1,200 years, it was actually one of the hottest and driest years in over a millennium, 2014 in particular. And then 2006 and 2011 were some of the wettest water years on record. So we can use those to ask, just do climatic drivers contribute to or outweigh or work with land use change in determining water quality and biological response? And then what can responses to these extreme years allow us to do or to say about future climate change impacts on our watershed? So to get at those questions, we took 3 basic approaches. First one is just looking at trends over time. Second one was looking at using ordinations to look at how some of our sites responded to these extreme years. An ordination is just shown on the right here, just a way of visualizing how different or similar sites are based on measured variables. So for example, a dot on that plot that's on the far right has higher air temperature, total coliform, and a dot on the farther left has a higher precipitation. And it also helps with variable reduction. So if we have two collinear variables like nitrate and phosphate, we can choose one as a proxy for the other. And this helps increase our power in some more complex models, which in our case, we took a structural equation modeling approach. And this is a causal modeling approach where we can actually look at what is the direct contribution of climate, land use, or both in determining changes in the last 20 years in our water quality and benthic response, and then use that to project out into the future. In our case, the climate data we used was from the California Basin Characterization Model, which just takes publicly available climate data and downscales it to California-specific watersheds at a higher resolution. So looking at just the trend analysis first, this is just looking at the climate variables, all kind of put on the same scale. And really this was just to look at what's going on in our extreme years. So on the left, we have years in blue are wet water year records, and years in red are critically dry. And if we just overlay these on the plot and look at the dark blue line, which is precipitation, the kind of darker purple line, which is aridity, climatic water deficit, and the dark orange line, which is our canopy cover, a couple things stand out that we have to consider for later on. Basically, canopy cover seems to respond well to increases in precipitation within the same year, but actually the relative decline in canopy cover that results from drought is a lagged effect. So it takes about a year for canopy cover to decline following extremely dry years. And then also, again, we're seeing climatic water deficit is decoupled from precipitation. It's more driven by temperature than precipitation. So we kind of have to consider all of these separate determinants of how our biological community is responding relative to climate-driven shifts in water quality. And then there's a lot to look at on this figure, but this is an ordination where we're bounding all of our sites within a given year, or all of our observations within a given year at one site within one colored polygon. So one polygon represents January through December, all of the measurements at a given site. So if it's smaller, there's less variance over the year. If it's larger, there's more variance. And if they're on top of each other, then they're more similar to each other, all of our sites. And if they're farther apart, then there's more variance in our sites. And really this was just to see, again, in our extreme years, in a wet year, all of our polygons are on top of each other, not much variance between sites. When we look at our extreme year, 2014, we start to see a lot more divergence among our sites. Just does show that yes, our sites are responding to extreme water years, primarily on the drought side and less so on the wet side. Another way to look at this is just the size of kind of the average polygons outlining all of our sites. And we see, again, our critically dry years. They're much larger, much more variance than our wet years. So all of this was preliminary stuff that went into the construction of our structural equation models. So we used the ordinations to focus on the best variables that were proxies for other variables, and then also those which varied a lot across these extreme years. And this is a schematic of what's going on in the background of a structural equation model, a little key on the bottom to help kind of walk your way through it. Where the circles are what we call latent variables. These are kind of these abstractions that we're trying to measure directly with the things in these square boxes. So climate in this case was a function of aridity, precipitation, and maximum temperature. Land use of percent impervious surfaces, canopy cover, catchment area, and percent forest change over the 16-year period in this case. And the best water quality predictors are listed on the bottom there. So what I'm going to show you is the best fit models, where the models in this case are testing the yellow lines here. So it's saying climate predicts water quality, land use predicts water quality, the dashed lines, so other candidate models, so climate influences land change, which then influences water quality. And what's going to be left on the screen are just these significant variables and relationships from the best fit models after testing all of those candidates. So for just the water quality model, the green lines here are positive relationships, red are negative, size of the arrow is the size of the relationship. Starting at the top left, we see, unsurprisingly, increasing aridity over the last 20 years, along with decreasing precipitation. And that was associated with this increase in this thing called water quality. That increase is kind of a subjective thing here, because actually what we saw was a collapsing of dissolved oxygen, a dramatic increase in water temperature, and then a slight increase in pH and conductivity. And then we also saw declining canopy cover along the riparian corridor, and actually increasing coniferous forest cover at the catchment scale were associated with these same changes in water quality. So then we took this base model and added in our benthic macroinvertebrate data into a model like this. And the biological data we used was a family level index of biotic integrity, or IBI, which is a single index of score of our stream health. We know IBIs have kind of fallen out of favor, and I'll come back to that a little bit later. And then we also use percent tolerant and percent intolerant taxa, and then tested this kind of tangled web of other candidate models to see basically how all these things are influencing each other. And the best fit model here, interestingly, was pretty much the same model as the water quality only model, with the exception of pH and conductivity actually dropping out. So there were not significant predictors of the change in the biological community. And then we did see a direct effect this time of canopy cover on water temperature. So this shows kind of that shading effect. So we're seeing a loss of canopy cover. It's just going to increase stream temperatures because they're just getting more sunlight. And so all of these things on the screen were then associated with significant declines in our index of biotic integrity and in our percent intolerant taxa. So increasing aridity, decreasing canopy cover associated with a collapse in water quality and biological communities over the last 20 years. So all of that was kind of looking at the what changed and the how, and then we wanted to then expand off of this and try to predict that into the future, what might happen across our watershed. So I'm going to show you here is again each of our sites and catchments, and then I'm going to overlay the projected increase in climatic water deficit, which is an anomaly, so departure from average at each of our sites. In this case, we're using 6 of the global circulation models that seem to match up really well with California projections and California data, and are also ordered kind of out of order, but from least severe projections on the left to most severe on the right. There's a couple out of order there though. And so what we see is in our upper watershed sites, they're actually pretty stable in terms of projected aridity in the next 100 years. In our mid watershed sites, we actually see some declines. And in our lower watershed sites, we see dramatic increases in climatic water deficit. We can then take this, plug it back into our model, and see what our changes in projected water quality may be. So those 4 variables that came out of our model, conductivity, dissolved oxygen, pH, and temperature, pretty stable at our upper elevation sites. But our lower elevation sites, pretty marked increases in conductivity, pH, and temperature, and a collapse in dissolved oxygen. Another way to look at this was to overlay our historical climatic water deficit variability and peak climatic water deficit. So warmer color means hotter in the past, larger means more variable. And this was useful for us to see how it wasn't just an elevation effect. So there are sites lower in the watershed that are actually more stable and cooler than sites in the upper watershed, and some sites right around downtown Nevada City in the middle that are both really variable and actually have gotten pretty arid in our 20-year record. But then when we overlay our projected increases in climatic water deficit, we start to get an idea of maybe some different sites for different management techniques. So our lower elevation watershed sites are both highly variable and projected to increase dramatically in climatic water deficit. So they're going to need the most attention in terms of adding in climate refugia, building in some additional resilience. Meanwhile, those mid-elevation sites that are highly variable are projected to decline in aridity. So maybe management there could focus on kind of preservation and maintenance of any climate refugia that's already there, and maintaining kind of that canopy cover, which we might be seeing as more like a seasonal variability effect rather than an increase over time. So there's a couple limitations with this model. It was just like a proof of concept. The biggest limitation is the structural equation model, which is based on linear relationships. We want to take a different modeling approach with some nonlinear relationships in the future. We want to incorporate physical habitat because we know that microhabitat can dramatically influence climate refugia. Like I said, we used IBI scores. We do want to use the California Stream Condition Index scores in future runs, compare those to ours, and see what we get. And then finally, some of you may have noticed we used primarily climate projections and not land use projections for the current version. This is because land cover projections are a little bit more uncertain. So our next step is to incorporate a lot of that uncertainty by taking kind of the extreme ends of change in forest cover, high end, low end, and then simulate what might happen if land cover goes in that direction. The last point I wanted to come back to was this weird relationship between NDVI and catchment scale forest cover. And what we think we actually are seeing here is a little bit of a fire effect, as well as just the different measurement scales. A lot of our sites are in the lower elevation watershed. And on the top left image, we have our watershed in red and the burn scar of the 1988 49er fire in white. And we see that by 2001, it was mostly broadleaf shrub cover and a little bit of woodland. And then by 2016, 30 years after the fire, it had recovered and started to transition to coniferous forest. Coniferous forest actually has a lower leaf area and a lower canopy coverage than a lot of the broadleaf coverage that we might be getting by the stream within that fire scar. So we have to dig a little bit more into this, but we think we might be able to kind of use this tool to also look at fire data moving forward. So with all of that, I would like to thank 20 years wonderful volunteers. I think we're actually approaching our 2-decade point coming up where we hit it this year. our water quality crew, bug crew, and all of the folks listed on this slide. And so with that, I will take any questions.
All right, thank you, Jeff. While everyone thinks about what questions they want to put into Slack for Jeff, let me ask one of mine. So Jeff, a lot of people have looked at the effects of land use and climate change and actually trying to predict the relative effects of each of those. Generally tend to show that land use really has more of an effect than climate change will. But you're kind of showing the opposite. What do you think is causing that?
I actually think it's probably more of just a watershed specific thing. Our watershed over our entire sampling period hasn't, to be honest, changed much in terms of vegetative cover and land use beyond the fire scar. There's some agricultural use lower in the watershed, but that's been pretty consistent over time. Yeah, a lot of it I think is just a lack of consistent change in land cover. So it's probably driving what's there, but maybe not necessarily the change because the land cover just hasn't changed much.
Okay. Do you think that the fires you're talking about were incorporated in your land cover measurements, or?
Well, in our case, they weren't. We were just looking at vegetative cover. So we were looking at, yeah, it was basically just shrub cover or conifer forest cover, which we know lines up with the fire scar. We did not incorporate anything like burn severity or any of that kind of stuff in this version of the analysis yet. Yeah.
Okay. Are there other questions for Jeff? Well, people are thinking a bit more. One last one I have is, I was actually a little surprised to see that in your ordination that the water quality seemed to be orthogonal to precipitation. Normally I see precipitation, the dilution of precipitation tends to move water quality around a little bit. Any insight of what's going on there?
Honestly, not really. We had thought about that same thing. We want to incorporate things like flow and precipitation more directly in some other future analyses. But I mean, it could just be that our stream, we do have 3 regulated dams on the stream. So some sites are going to be much more sensitive to precipitation. And then some of our downstream sites, after really extreme precipitation events, are going to see some pretty dramatic responses to those precipitation events. But I can't really comment more on what else is going on there without digging a little bit more. Could just be that, again, the nature of where our sites are relative to dams and things.
Right.
Yeah.
Okay. Actually, so Jeff, I understand there's one more slide you want to present.
Oh, yeah. Let it go forward. Okay. Yeah. So unfortunately, just wanted to let everyone here know, Sierra Streams Institute, we did unfortunately lose our office in the Jones fire in August of this year. So we are sharing this GoFundMe information to basically talk about what happened, things that we're looking for in terms of support. Want to give a really quick shout out to Eric Burrs for some really great help on replacing some of our instrumentation that was lost in the fire, both with temporary and long-term replacements. But basically, we are looking for any monetary support people are able to give through the GoFundMe here. Instrumentation support. So if you have old instruments that you're cycling out, we would love to take them on during our recovery and rebuild effort. And then finally, also just even collaborations. We have kind of a unique opportunity after losing our office and losing a bunch of our equipment to kind of recalibrate the way we sample and the way we operate. And so we're really interested in talking to folks that might be interested in working with the citizen science group that has our 20-year data set to try to come up with some new projects to work together and help us through our recovery. So if you have any questions about that or any ways you would like to help, please contact me via Slack or via email.
Excellent. Thank you, Jeff.
Thank you.
Go ahead, Allie.
Sorry, jumping in. Yeah, sorry to hear about that loss. It's really sad. I don't think the information's up on Slack yet, but we will work on getting that up there shortly. And at this point, I think we can break for lunch. I'll keep this information up here for a while so folks have access to it. And then we are coming back at, let's see, 12:10 p.m. for lunch. If you want to check out the posters, those are all still there. So pop over there. I'll put a slide up in a second that has that information. Otherwise, we'll see you guys back here at 12:10. And thank you, John, and the other speakers for that last session.
Thank you.
The recording has stopped. Good afternoon. Can anyone hear me out there in Zoom land? This is Mark.
I can hear you, Mark.
Hey, Mark, how are you?
Hey Keith.
Yeah, hi Keith. Hi Mark. Hi Marks, and this is Keith. I'll be moderating the session.
Great, thank you. Do the presenters have control over the slides, like forwarding the slides?
I believe so. That is that when Shuka jumps back on.
Yeah, she probably makes us host.
Yeah, I know yesterday with the England folks, they were having Shuka control it, but I think that was a cross-continental connection.
Nice. Okay. Mark Silos, I'll be going through my stuff pretty quickly to make sure you have time at the end for your case study.
Sounds good. And then I'll just probably have you keep the slides on, so I don't have to so you don't have to stop screen sharing and then I have to do it.
Yeah, no, we need all the time we can get.
Yeah.
I did a practice and you had like 3 minutes left. I'm like, uh-oh, I can't do that. So I'm hoping to give you 5.
Sounds good.
And then there is a delay between our conversation and what's going out to the webcast. So I'll have like at least 1 or 2 questions for each of the presenters to keep the conversation going while we wait for comments.
You can bleep out our bad language too.
That's right. You can share some strong words about other nutrient management philosophies, Mark.
Hey Keith, if I understand correctly, so will we be fielding questions in the Slack after the talk?
So what we're usually doing is Shuka will be helping them manage the Slack, and so will I. And so we'll have a few, depending on the timing of your talks, if there's still time available, we'll address some questions audibly. But then you guys can definitely, after you finish, go and see if there's any other issues or additional comments you'd like to make in the Slack afterwards.
Sounds good, thanks.
Mm-hmm.
And am I to share my screen and control the slideshow? I think that's what we're doing. I'm just wondering why we sent you in our PowerPoint, but anyway, we'll.
Hey guys, yeah, so we will be controlling the, we'll control the slides from here, or you can request control. It's up to you. So you send them to us because we have one master presentation deck over here.
You have a recommendation for the best approach?
You can try advancing on your own. Some people have been having issues with that. So if you're cool with playing next slide game, then we can.
Yeah, that's fine.
Okay, then let's do that.
Thank you.
Sure.
This meeting is being recorded.
All right, good afternoon, everybody. It's 12:10, so we're going to go ahead and get rolling back for our last session. This session is going to be moderated by Keith Gregson, who is a swamper at the California Water Boards. Keith is an environmental scientist, and he co-leads the Freshwater Harmful Algal Bloom program, which is one of the programs under SWAMP in addition to the Bioassessment program. Keith received his PhD from UC Berkeley in 2017, and he looked at toxic cyanobacteria and algal ecology in Northern California. And Keith joined the water boards, what is that, Keith, in 2019? I feel like it's been longer, but yeah, 2019. And with that, I'm going to hand it off to you.
Great. Thank you, Allie, and thank you, everybody. I hope you had a nice lunch. So I'll be moderating this next session, and we'll have 3 speakers. And after each speaker, we'll have a brief time for some questions, and there won't be a panel discussion at the end, as in some of the other sessions. But please be active on Slack and posing your questions there, and the conversation can continue on that platform. So our first presentation is by 2 presenters, Mark Butel and Mark Selos. Mark Butel is a professor of civil and environmental engineering at UC Merced. His work focuses on sustainable control of dilute pollutants in the environment, including the effects of bottom water oxygen addition on mercury cycling and bioaccumulation in lakes and reservoirs. And Mark Selos is an associate water resources specialist with Valley Water in San Jose. He has a bachelor's of science in environmental geology from UC Santa Cruz and is currently pursuing a PhD in environmental systems at UC Merced. He is a certified lake manager and serves on the executive board of the California Lake Management Society. And their talk is titled Hypolimnetic Oxygenation to Control Mercury Bioaccumulation in Lakes, 2 Case Studies.
Okay, yeah, could you guys please control the slideshow as we had discussed, and I can just give you a verbal on moving it forward? Thank you. Okay, so we've got quite a bit to cover here in our short 15 minutes, so I'm going to keep a brisk pace here. Mark and I are going to tag team here. He'll finish it up, but we're going to do an introduction to lake oxygenation, what that's all about, a bit of a dialogue about mercury cycling in aquatic ecosystems, in particular lakes. Then we have 2 case studies to talk about, Twin Lakes, Washington. I'll talk about that one, and Valley Water's efforts here in the Bay Area, and then some conclusions. So next slide, please. Okay, you can check out these two studies in a 2014 publication I did on Twin Lakes, and Mark has got one in review in environmental pollution. So look out for those if you want to dig deeper into these two topics or two case studies. Thanks. Next slide. What is lake oxygenation? It's the engineered addition of dissolved oxygen to bottom of lakes and reservoirs using pure oxygen gas rather than air as an oxygen source. So this is the real paradigm shift. We're not using air, we're using oxygen gas. And of course, oxygen gas has 5 times the oxygen as air. So your engineered systems can get very small. This was pioneered by Richard Spiess, an emeritus professor at Vanderbilt, and there's this great quote, "Air is free, but oxygen is cheaper." You can really get a lot of oxygen into lakes and reservoirs using pure oxygen gas. Next. There's 3 main systems that you can use, and we're talking about deeper reservoirs here that are thermally stratified. There's a bubble plume system where you set out coarse bubbles. There's the line diffuser system where you weep in tiny bubbles. Those are the 2 systems that we'll look at today. Then there's the Spiess cone where you have a submerged pump and you can suck water in and then put the water in contact with oxygen gas inside the cone, and out comes your oxygenated water. What's great about these systems is for the most part, they maintain thermal stratification, so you get a nice, cool, oxygenated pocket of water down in the bottom of your reservoir. Next. We're going to look at each of these 3 very briefly, just with some pictures. So next slide. So the Swiss were the ones who pioneered the deep oxygenation injection. And so what we see here is Lake Baldag, a deep lake, lots of ag around it. And down in the bottom right-hand corner, you see the oxygen system. So this is going to let out large bubbles. The water column's deep enough that a lot of that oxygen dissolves into the water column. The bottom is the onshore storage of the liquid oxygen, which you convert to gas just by warming it up, and then the gas goes down to this diffuser, which would be at the bottom of the lake. You can produce oxygen onshore. In fact, the Valley Water case study does its own onshore production using electricity. The Twin Lakes, you'll see, has the liquid oxygen storage. Next. This is another system. It's the line diffuser system where you have linear diffusers, sometimes miles of them, and you weep out small bubbles out of porous hose. This was developed at the Tennessee Valley Authority. Douglas Dam, they put in 90,000 kilograms per day, just an enormous amount of oxygen. In this case, they're getting oxygen into these huge hydroelectric releases to maintain oxygenated conditions in the release waters and tailwater area. Next. The Spiess cone, a good example of that is Comanche Reservoir, and there was a recent publications on this in Lake and Reservoir Management Journal. Here we have the cone in the middle being launched on the red barge. You can see it's about 25 feet tall. That's going to sit down at the bottom of the reservoir. On the left is the diffuser, which also will be set on the bottom to squirt the water out into the reservoir. And very small, modest footprint down in the lower right, you've got your liquid oxygen tank and your evaporator. So amazingly small footprint to be able to get 8 tons of oxygen per day into your system. In this case, there was a fish hatchery just downstream that was in need of oxygenated water from the reservoir. Next. So those are 3 case studies. Now let's talk about mercury. I'm sure a lot of you know about the mercury cycle, but you can let's just go over it. And really the issue is, how is it linked to oxygen? And so you can have atmospheric deposition of mercury if you follow that central gray arrow down. In the case of Valley Water, you can have legacy mining inputs of mercury. So we have our input of Hg2 in this middle circle. That Hg2 can get converted to methyl mercury by anaerobic bacteria. The methyl mercury is the CH3Hg down in the right corner. It can be demethylated too, but the bottom line is this this methylation process is done by sulfate reducing bacteria and other anaerobic bacteria. The methyl mercury is the form of the mercury that gets up the food web. So now you see this linkage between oxygen and methyl mercury production. If we can add oxygen to aquatic systems, in this case, the profundal zone of lakes and reservoirs, can we repress this methyl mercury production by basically robbing the anaerobic bacteria of their livelihood? Next. So let me talk about the Twin Lakes case study. I worked at Twin Lakes as a professor up at Washington State University before I came down to Merced. And so really, really interesting system. It's on the Colville Indian Reservation. And so this picture to the right is showing you why they wanted to add the oxygen. The bars are where trout are in the water column. And they were trying to bring back a native Lahontan cutthroat trout population in these lakes. And so what we have is the circles, you can see it is the oxygen. So as you go down the water column, there's no oxygen in the bottom. The square is the temperature, warm on top, cold on the bottom. So you can see the trout are in this classic DO temperature squeeze where it's too warm on top, but no oxygen on the bottom. And this obviously is not great habitat. So the tribe wanted to do a pilot study to oxygenate the lake and see if that helped the trout. The mercury was a curiosity and an add-on of mine from a research perspective. Next slide. So what was neat about this study is we had this two lakes, twin lakes, right? So the North Lake had the line diffuser in the deep basin. The South Lake was used as a reference. You can see in the upper system, there's a oxygen, they rented an oxygen tank on a trailer. That's how easy it can be. Throw up a chain link fence. On the left, this black tube is what goes down to the lake. So incredibly modest infrastructure to get an ecosystem response of enhanced oxygen. The bottom picture there is the line diffuser, which is being put into the lake. It will ultimately be sunk down to the bottom. Next. So here are some isoplasts for South and North. South is on the top where we did not add oxygen. North is on the bottom where we started oxygen in 09, 10, 11, 12 as well, but we don't have that data on this figure. So you can see for the upper two panels, there's that red blob of anoxia, right? These aren't especially deep lakes, around 15, 16 meters. They're bowl-shaped, and boy, that bottom water goes anoxic very quickly. And coupled with that anoxia, you can see is this dark buildup of methyl mercury. The concentrations aren't extreme. Mark Selos would just scoff and chuckle at these tiny concentrations with the systems he work on. But sure enough, you really see this connection between anoxia and methyl mercury buildup in the profundal zone. Now in Twin Lakes, we started adding oxygen in 2009, and you can see the response, less red. And then the bottom panel, sure enough, we saw less methyl mercury. If you look at the 2008 panel, you see the red in North Twin and then the red decrease and that yellow kind of increase. That was when they tested the oxygenation system. In fact, we have a paper out on that, but we saw this very rapid response. And then from year to year, we saw a good response. What we did see also is in years where they tried to save money and not add so much oxygen, like in 2010, you see a bit of anoxia and some methyl mercury. And in fact, 2012 was similar to 2010. Not so much oxygen, more methyl mercury. So real tight linkage here between status of the oxygen condition in the bottom and methyl mercury. Next slide. So this was the big surprise. When we had in the North Twin where we added oxygen, we saw more mercury in the zooplankton. And you can see that in this simple bar chart. The lower one is South Twin where we did not add oxygen, and in North Twin Lake, sure enough, we perturbed the system and enhanced mercury uptake. And the same thing in the fish. These aren't like dramatic levels in fish, but they went up 10%. And you know, fish data sets are tough to work with, and these are different cohorts of fish. But in particular, if we look at the Eastern brook trout spring 2010 stocking, It appears to have gone up from 50 to 75. Of course, the fish conditions changing too, so that's kind of an interesting covariate. But anyway, we saw an increase in mercury in bioaccumulation, basically. If you want to understand the mechanisms, check out the paper. Next slide. So what was interesting is in North Twin Lake, where we did add the oxygen, there appeared to be a pretty tight linkage between oxygen status of the bottom waters and the methylmercury in the zooplankton. I noted earlier that in 2010 and 2012, they didn't add oxygen as robustly in the other years. And sure enough, if we look at mean annual hypolymnetic DO and mean annual zooplankton, we saw this interesting trend. So it's kind of odd. We added oxygen. It seemed to the reference lake increased bioaccumulation, yet we did see this connection between oxygen and mercury bioaccumulation in North Twin. And I think we're handing it off to Mark Selos now. Next slide.
All right, thanks, Mark. So I'm going to kind of be going over an entirely different system that's much more polluted than the one that Mark was talking about. And I'm going to be really briefly discussing the results of a 15-year study. And so if you're interested in learning more about this, I'm actually going to be giving a presentation next Thursday at the California Lake Management Society online conference. And that's free registration. You can check it out at calms2020.org if you want to learn more. So the study reservoirs that I'm working on are polluted by the former New Almaden mining district. It was North America's largest historic mercury mine. And about the Titanic's worth of weight of mercury was actually extracted from this hill. So it's a really highly disturbed, highly polluted area, and we have sediment mercury concentrations that are measured in the reservoirs on the order of parts per million, which for mercury is actually a lot. So you can go to the next slide. So just like in Mark's study reservoirs, we kind of saw this really predictable seasonal pattern of methylmercury production that corresponded with anoxia and the hypolimnion. So on the left here, you can see this is just one typical year at Guadalupe Reservoir, where around from like June to November, you can see that the reservoir was thermally stratified, and that corresponded with depletion of oxygen in the bottom water. And you can see at the bottom plot on the left, that was when we saw most of our methylmercury produced, up to like 25 nanograms per liter, parts per trillion, which for methylmercury is really a lot. And one of the things that we noticed was that if you look at the right side, in the middle graph, this is our sulfate in the bottom water. And we saw our methylmercury spikes at the bottom corresponding with the depletion of sulfate. And so a conclusion from that is that it's mostly as a result of sulfate reducing bacteria that are the ones that are primarily implicated for producing methylmercury. Next slide. So we have some pretty high fish tissue mercury concentrations in these reservoirs. On the left side of the plot, you can kind of ignore the x-axis and focus on just the standardized 350 millimeter mercury concentrations. The ones that are in blue are the reservoirs that we manage at Valley Water. And you can see these are kind of like high outliers in the state. On the right side, you can see fish from like 30 centimeters to 50 centimeters have mercury concentrations from like 5 to 10 milligrams per kilogram. That's parts per million. That's really a lot, assuming that most of that is methylmercury. And I kind of did a back of the envelope calculation. And basically, if you eat 1 8-ounce serving of bass from Guadalupe Reservoir, it's the mercury equivalent of like 38 cans of tuna. And so these are really, really highly polluted systems. Next. So we use the same kind of hypolimnetic oxygenation systems that Professor Butel described in the Twin Lake study. And it's a line diffuser system. And one of the things that we were really pleased to see is that even though the diffuser was limited to the very deepest part of the reservoir, it actually was able to oxygenate the entire sediment water interface in the entire reservoir. And this is kind of what this plot is showing. The red is the actual diffuser line, and the 0 is basically the inlet of the reservoir, and the 1,000 meters is like the dam face. So you can kind of see we did fully oxygenate the entire reservoir. Next. So these are some of our results for mercury in methylmercury in water. We oxygenated 3 reservoirs. The one that's shown in purple on the left is Stevens Creek Reservoir, which was not actually mine impacted, but is impaired for mercury. And kind of the main result that we saw from this is that we saw these large reductions in bottom water methyl mercury shown in the bottom center of the left plot. You could see like in Guadalupe Reservoir in particular, mean concentrations went from around 12 nanograms per liter to around 1 or 2. And so that was a big reduction. But we also didn't see any reduction anywhere else in the reservoir. So like right above that plot, you can see we didn't see any significant changes in surface water methyl mercury, except for in Stevens Creek where it actually increased slightly. And this is really a concern because it's thought that the methyl mercury that's up in the surface water in the photic zone is where it enters into the food web and is able to bioaccumulate. So theoretically, without reducing methyl mercury in the photic zone, you're not actually going to reduce it in fish. And on the right side, this is kind of like average water column concentration that we measured methyl mercury in 5 depths throughout the water column and sort of considered the whole water volume and took an average. And we actually didn't see any significant differences in average water column. And so all of the difference that we saw in the methyl mercury seemed to be only in bottom waters. Next slide. And so basically our running theory is that the hypolimnetic oxygenation systems are mixing profundal waters up into the photic zone. And we saw evidence for that in many ways that I don't really have time to describe right now, but we saw either unchanged or increased methyl mercury in the photic zone. We saw increased nutrients, and we actually saw algae blooms. And so we think that the increased primary productivity in the photic zone actually caused a bloom dilution of methyl mercury throughout the food web, which could have led to these 2 reductions that we saw. We have declining trends in Guadalupe Reservoir and in Stevens Creek Reservoir. In Almaden and Calera, we didn't really see any changes. Next slide. So we're just about out of time, but so both of these studies, we saw like a strong linkage between oxygen concentrations and methyl mercury. However, we did notice that when you put an oxygenation system in a reservoir, you're not just changing a certain process, you're changing the entire system. And unfortunately, these perturbations led to things like, in Mark's study, higher bioaccumulation, in my study, higher nutrients in the photic zone. And yeah, if you want to learn more about that, check out my presentation next week at the California Lake Management Society, columns2020.org. Thanks.
Great, thank you, Mark. And Mark, I just had one quick question. Could you talk a bit about what the half-life of the methylated mercury is and what its fates are apart from getting picked up in the food web? Does it just persist in the water if nothing consumes it, or does it eventually degrade into a less bioavailable form?
Yeah, so there are a couple things that can happen to it. So it can be demethylated either by light. So there's photodemethylation, which is typically kinetically a really slow process. And there's also microbial demethylation. So some of the microbes that methylate mercury can demethylate them, or demethylate methyl mercury. And then, so what we typically see is like you get a lot of methyl mercury production in this small redox window. And once it gets too reducing or too oxidizing, then we kind of see demethylation predominate. And so it's a super dynamic system, but we do think of the biota as kind of like a time integrated sponge for methyl mercury where it's not really going to be transforming that much.
Wow.
Yeah, and I can add to that. In Twin Lakes, there is this synergy between sulfide and methyl mercury, and sulfide will stick to the methyl mercury and keep it in dissolved form. And that was one of our thinkings for why we saw less kind of pretty low bioaccumulation in South Twin. Is sure, we had this real buildup of methyl mercury in that bottom water column, but it was all bound up, dissolved, but forming partitioning to sulfide, and so there it was dissolved but unavailable to kind of get into the food web. And once we oxygenated the lake, what mercury was around was no longer linked to that sulfide and able to get into the food web more. So there's a lot that can go on. Just because you're anoxic doesn't mean you're, like Mark said, there's a kind of this modest redox window where you get this methylation and the methyl mercury appears to be more conducive to getting into the food web. In Twin, we had a really sour, stinky bottom water, a lot of methyl mercury, but it didn't seem to get up into the food web much.
Wow. Great. Well, thank you for that extra information. And thank you both for presenting. We are out of time on this session now, or on this talk now. So please, if you have additional questions, post them in the chat and or on Slack, and Mark will be monitoring that. And so our next speaker is Karen Atkins, and she is interested in understanding how biological, chemical, and physical factors affect aquatic ecosystems. She's a PhD candidate at UC Davis, working to understand what factors contribute to benthic algal blooms in pristine oligotrophic lake habitats. And most recently, she's using 28 years of field data collected from Lake Tahoe and statistical modeling techniques to identify the environmental conditions that control paraphytic algae biomass. And understanding Lake Tahoe's unprecedented data set will shed light on the trends of increased benthic algal blooms in lakes globally. And her talk is titled, Statistically Modeling 28 Years of Periphyton Blooms in an Oligotrophic Lake. So welcome, Karen, and take it away.
Thanks, Keith. Can everyone hear me? I think so. So I changed my title just a bit, but it's the same general idea. Let's first talk about what periphyton is. Periphyton is this kind of fluffy algae that you see in the picture on my slide. It's actually microbial growth on substrata, and sometimes it's called attached algae. So this periphyton is really important in ecosystems. It serves as the base of aquatic ecosystems in many cases at natural levels. Next slide. But you can have too much of a good thing. At high levels, periphyton can be an early indicator of ecosystem change. And we're seeing these periphyton blooms in pristine lakes all across the world. This map here shows in the dark red and the dark green color here, those are areas where we tend to see more periphyton blooms. And these lakes on the bottom are some examples where we've seen anecdotal evidence of increasing periphyton in the near shore of lakes. I say anecdotal because we actually don't have the data sets that would be required to prove that this is really happening across the globe. And that's because people haven't really monitored the near shore of lakes, and because these data sets that we have are just starting to emerge with long-term data sets that would be able to prove this. However, in Lake Tahoe, and next slide, we have a really unique data set that enables us to look at the change over time in periphyton. I'm sure many of you, being from California, are familiar with Lake Tahoe on the corner of California and Nevada. It's really, really deep, and it's got quite a long shoreline of 116 kilometers around. It's also oligotrophic, so it has very few nutrients. In 1950s through 60s, there was a lot of development that was happening in this area. There were the casinos that we see today, the ski slopes were being put in, and we also had a lot of vacation homes that were going in around this time. Then in the 1960s, Charles Goldman was the first person to write down and observe that there were nuisance levels of periphyton happening in Lake Tahoe. And then in the 70s and 80s, we got some of the initial periphyton investigations that were starting to study what are causing and what the trends were in periphyton. Then in 1982, the data set that I will be presenting about today began, and we started regularly monitoring all the way through today with a couple data gaps. Currently, many of the agencies in Tahoe have been receiving complaints that the periphyton is increasing to levels people hadn't seen before. And so that brought us to the next slide, to some of the research questions that will address kind of both the global issues as well as some of the local issues. So the questions that we wanted to ask were, has periphyton growth increased since regularly monitoring began in the 80s? When was the peak biomass around the lake every year? And where is the most periphyton? As well as, does the type of periphyton at different lake levels affect how much biomass we see? Next slide. So let me tell you guys about the field methods that we've been using. One important thing is that since the beginning of this research in 82, we're using a 0.5 meter depth. So as you see the lake level go up and down, our location of study also follows that and goes up and down. We have been monitoring on 2 different time scales. One we call our routine time scale, which happens at 9 sites around the lake and ranges from 3 to 15 times per year, roughly monthly, focused on measuring around the peak biomass time in the spring. In addition to that, we've been doing the synoptic monitoring, which happens at around 54 sites. So it's a lot of sites around the lake. And that's been happening since 2003 and is done once per year on the spring peak. In addition to that, at each site, we monitor for a couple different constituents. We monitor ash-free dry mass, which is basically a measure of the carbon in the periphyton. We measure chlorophyll A, and we measure percent cover. Next slide. So in this slide, we see that ash-free dry mass is in the first plot here over time, and then below it, we see chlorophyll A over time, and finally, we see the number of samples collected each year over time. So in that last plot, I just want to point out that there's a lot of variation in how many samples were actually collected each year, all the way from no samples to over 100 samples. And there's also some pretty large data gaps in the beginning of this data set, especially in ash-free dry mass. And so I just wanted to point that out, keep that in mind as we move forward with the analysis of this data. Next slide. So one of the first things that people notice as they start looking at periphyton is that there's 3 major groups of periphyton in Lake Tahoe, or communities. And so the first one is a stock diatom community, and you can see the picture below. They look pretty different from one another. The genera, or some of the genera that make up this stock diatom community, is the Gomphoneus and the Gompphonema. In addition to that, filamentous green algae is also in Tahoe, and that community is made up of algae such as Mugiodia and Cladophora. And finally, there's a cyanobacteria community that occurs a little bit deeper in the lake generally, and is made up of Calothrix and Nostoc as examples. Next slide, please. So now let's get into some of the trends that we were seeing. So we did a Mann-Kendall test, or quite a few Mann-Kendall tests, to evaluate trends over time in the periphyton. So first, if we look at this blue box on the top, you'll see that if we combine all the years together and we look at chlorophyll A, there was no significant trend in periphyton, up or down. However, if you do the same analysis, but for ash-free dry mass, there's a negative trend. So that was actually the opposite of what we thought we would find, which was surprising. Then if you break it down by the dominant community for each year and just use those years, and you look at cyanobacteria, we had a significant negative trend again, but with the diatoms, there was no trend. So a lot of that negative trend is happening within the cyanobacteria community. Next slide, please. Now, we also did some Kruskal-Wallis tests followed with Dunn tests to look at the monthly variation. And so in this first chart, we see ash-free dry mass for each month, and then the chlorophyll A for each month. In the winter and kind of early springtime, in this red box, we see the highest biomass. And then the lowest biomass is occurring in the summer. So if we look at the typical Tahoe annual lake cycle, lake level cycle, that highest biomass is occurring during the increasing lake level of Lake Tahoe, which kind of makes sense because you'd expect as you're getting a lot of water coming in, you're probably getting a lot of nutrients as well. Then the lowest biomass is actually happening when we're at the top of our lake level. Next slide, please. Okay. So now let's talk about community and lake level and how that's really tied together. The first chart that we see here is lake level over time, and then we have the year. And then this next chart here is looking at the annual proportion of observations where each periphyton community occurred, and then the year again. So the first thing that you'll kind of notice is that there was no year in which filamentous green algae were the dominant community that we saw. Now, if you look at the plot on the left, you'll see cyanobacteria highlighted, the cyanobacteria dominated years highlighted in red. And those tended to occur when we had either a declining lake level or a low lake level. And then the stock diatoms on the right were occurring at the highest of lake levels. So lake level was clearly affected what division we were seeing at 0.5 meters. And what was really interesting too was that when we looked at what the biomasses were for each community, the cyanobacteria dominated years where had higher biomass maxes than the stock diatoms dominated years. And so this clearly has a lot of implications, and one of which is that the drought cycles that we see from year to year in California is really controlling in some ways the biomass that we're seeing. The years that we did this research on had a lot of drought cycles compared to, for example, the 40 years before it. Next slide, please. Okay, now let's talk about the spatial variation. If we look at the medians of ash free dry mass, chlorophyll A, and percent cover again, you'll see that the north and the west sides of the lake had a lot more periphyton than the east and the south. And if you look at the maps on this slide, the dark green dots have higher biomass and the white dots have the lowest biomass. Again, for chlorophyll A, ash free dry mass, and percent cover. And so some of these trends make a lot of sense. So for example, nitrogen and phosphorus have higher loads on the north and west side of the lake. So that's kind of you'd expect to see more algae there. But in addition to that, the prevailing wind direction in Lake Tahoe is coming from the southwest into the northeast. And so what you have there is more waves happening, and therefore perhaps more shear stress on the rocks, and therefore more sloughing happening. In addition to that, in the southern portion of the lake, we have really sandy substrate. And when we've been snorkeling in that area, we've seen that a lot of the sand seems to get kicked up and it might settle on the periphyton, which kind of shades the periphyton and also acts as an abrasive on it, and therefore might reduce levels in the south. The last thing I want to point out about this slide is that there's a lot of variation just between sites, one next to another. Next slide, please. So given all of those trends, why are people reporting increased periphyton? So there's a couple reasons that we think this might be occurring. So first of all, perhaps we started monitoring too late and we didn't catch this baseline of algae. Another reason could be this drought cycle that I was talking about earlier. We have had frequent droughts over the last 3 decades, and maybe that changed what biomass was visible from shore, changing what people could see. But since we have this 0.5 meter variation in our, and we're following the lake level, perhaps we didn't capture it in our data. Another reason could be that there's a lot of invasive aquatic plants in Lake Tahoe, and there's metaphyton that may or may not be increasing. People are doing research on that now. But those could have been identified, misidentified as periphyton. Next slide. So zooming out onto a more global scale again, I just want to remind everyone that periphyton blooms are occurring in oligotrophic lakes across the world. And so it's really difficult to prove whether or not we have increasing periphyton unless we have data sets that are monitoring periphyton. And so we must begin monitoring the near shore now, and we have to do it, So it's crucial for understanding the causes of periphyton, and we can't really learn more about the periphyton blooms unless we're monitoring it. Next slide, please. think we skipped one. Okay, thanks. So this paper is that paper that I just talked about now is currently in review in Hydrobiologia. So check that out when it comes out. And I'm currently also diving deeper into this data set, and I'm working with some Bayesian hierarchical model methods, and I'm trying to use other data sets that we collect at Lake Tahoe to combine them and look at nutrient levels and lake levels, wave action, and water temperature, as well as light, and see how that all affects periphyton biomass at Tahoe to try to basically understand what is causing these blooms when they're occurring. Next slide. So here's the reference I used for my maps. And next. Any questions?
All right, thank you, Karen. We did have a question come through Slack. Somebody was asking about how the timing of the peak periphyton biomass relates to ice-off dates in Lake Tahoe, and maybe also more generally some of those that transition from winter to summer and the spring conditions.
Yeah, good question. We don't really get much ice in Tahoe. It never really freezes over all the way. So yeah, it's a little bit different conditions. I know that in places where there are full freeze overs of the lake, that there are often periphyton growing underneath the ice, depending on how clear it is, of course. Yeah.
Great, thanks. And I also had a question. You were saying that the overall trends were a little bit weak, but then you showed some of that spatial variation. Were there any spatial hotspots where you saw, a cluster of sites in particular regions of the lake that did show a stronger increase of chlorophyll or ash-free dry mass, or the inverse, a strong decrease, and that spatial variation was then dampening out the overall trend?
Yeah, we definitely did like a site-by-site analysis as well, and that's in the paper, but I didn't have time to talk about it here. Some of the sites that have really high hotspots are near the outflows of rivers. And so, yeah, those are obviously just bringing a lot of nutrients. And then some of the cities also had high hotspots near them. Yeah.
Okay, and then there's one last question again from the Slack channel. Yeah, is that the person's asking about the change in surface temperature over the study period, and if there's a trend in surface temperature, and if that might be impacting periphyton biomass.
Yeah, that's a great question. So there definitely has been an increase in average surface temperature at Tahoe over the time span due to climate change. And that is something that we're going to be looking at in my Bayesian model to see if that's influencing the periphyton.
Great. All right, wonderful. Well, I think we're out of time. So thank you, Karen, and we'll look forward to
thank you.
Your presentation next year on the Bayesian statistical modeling results. So
sounds good.
All right, and all right. So our last speaker is going to be Brian Furman, and Brian is an aquatic technology development scientist at the CPRO Corporation, working out of Sacramento, and is also the Northern California director for the California Lake Management Society, or CALMS. He received his PhD in environmental systems from UC Merced, where he studied redox and oxygen control on water quality in California reservoirs. He also holds a Bachelor's of Science in Chemistry and an MBA and MS in Environmental Engineering Sciences. He has previously worked as an instructor for an environmental engineering course on water quality and as the quality assurance manager at an environmental laboratory. Oh, and we're pulling up his slides now, so Brian, sit tight. Shuka's getting them. And Brian's talk will be managing the nitrogen to phosphorus ratio for improved aquatic ecosystem function. Great, and here come the slides.
Perfect. Thank you, Keith.
Thank you, Brian.
Really great conference. I'm excited to be a part of it. So yeah, my talk will be today on managing the nitrogen to phosphorus ratio and how that could help improve an aquatic ecosystem. So first, next slide. I'd like to just give a shout out to everyone that's helped me along the way. So Dr. Mark Butel, you probably caught his presentation just a couple slides ago. That was my PhD advisor, great guy, does some really interesting research. Everyone over at UC Davis and the Tahoe Environmental Research Center, or the Turk Center. So a bunch of really great researchers over there working on Clear Lake. And so you can catch some of their work. There's a great poster, if you have a chance to check it out, by Nick Fremstead. And so I'll go into some of the work they've been helping me with. And then, of course, everybody at CPRO. So Dr. Wes Bishop, AJ Jones, Dr. Mark Hauman, Mike Pierce, Scott Schuler. Next slide. So when I'm talking about aquatic ecosystem function, what I'm really getting at is, you know, the transition between oligotrophic, mesotrophic, and eutrophic. And so oligotrophic is kind of like a Lake Tahoe, like crystal clear blue water, very low algae abundance, very low organic matter in your sediment. Whereas on the opposite spectrum, you're going to have a eutrophic lake or aquatic system that's going to have just a ton of algae everywhere, really green. If you get a cup of that water, it'll look like some Kool-Aid, some lime green Kool-Aid, and then you're going to have this really kind of nasty muck with a lot of, you know, black stuff in it and partially degraded organic matter. And so naturally there are some eutrophic lakes, but in general, most lakes and aquatic systems are naturally oligotrophic or mesotrophic. And so human activity tends to lead to eutrophication of these. And that's primarily just driven by us putting nutrients into these systems. Next slide. And so the problem with eutrophication is it's really this kind of positive feedback loop upon itself, where a lot of algae will grow and it's too much for the food web to actually eat everything. And so that algae will die and it will sink down and hit the sediment water interface. And so that stimulates a lot of bacterial respiration. And so in that process, the redox ladder, as they call it, is really important, where all the microbes are going to be using the most favorable redox acceptors first. So they're going to be using things like oxygen, and then they're going to go down and use nitrate and use manganese and use iron and use sulfate and use carbon dioxide for methanogenesis. And the further you go down that redox ladder, the more likely you are to release nutrients. So the release of phosphorus is associated with iron reduction, which is about halfway down that ladder. So really trying to keep that redox up at a higher level, as you know, Mark, Silos, and Butel were talking about with hypolimnetic oxygenation, that has a lot of beneficial aspects for water quality. So when you have all this organic matter that hits the sediment, and you drive the redox down, you're going to release all of these nutrients, grow more algae, and it's going to be this vicious cycle that just keeps going. And you can lead to stuff like bioaccumulative methyl mercury production, releasing sulfide gas. Next slide. So as I mentioned, the aquatic food web is really important because it kind of keeps this in check, and there's a system of checks and balances. So if you have a ton of nutrients, which is at the bottom of that little chart, then you're naturally just going to grow a lot of phytoplankton or algae. And so a way that this food web can keep itself in check is that if you have enough zooplankton, for instance, to eat all of this algae, then you can kind of, you know, keep the system from going out of whack. And, you know, if you have enough consumers of the algae, then you're not going to have as much of that that's going to die and hit the sediment water interface. And so sometimes there's also other feedback loops. So say if you have too many small fish that eat zooplankton, well, then you wouldn't have a big enough zooplankton population to actually keep that algae in check. So some people have stocked like big game fish that will keep the small fish in check and allow that zooplankton community to thrive and to eat that algae. So starting at the base is the simplest theoretically, you know, just let's have less nutrients, but feasibly and realistically, that's a very difficult concept to accomplish. Next slide. And so I was kind of simplifying when I was saying phytoplankton and algae and zooplankton eat them, because there's actually different kinds of algae. So blue-green algae, I'm going to call them fake algae because they're not eukaryotes, they're prokaryotes. So they're not actually related to green algae and the other algae that evolved later on. And so I'm going to refer to those as like the real algae, and they're beneficial. So zooplankton, at the end of the day, zooplankton would much rather eat some green algae or some diatoms or the other real algae species. And cyanobacteria or blue-green algae, the fake ones, they produce a lot of toxins, and it's not actually for humans. I mean, it's bad. Like these harmful algal blooms are toxic to humans, to wildlife, to everyone else. But the reason why they do this is to avoid being eaten. So zooplankton really don't like to eat cyanobacteria. And this causes them to be able to live out their entire life cycle, which is only a couple weeks, and then they'll die and they'll sink to the bottom. So whereas a green algae might be a good food source for some zooplankton, and it might not actually get to the point of dying and sinking to the bottom and adding to that organic matter of the sediment, cyanobacteria unfortunately aren't going to do that. And so the one thing that we want to take advantage of with this N to P ratio is the fact that there's a decent amount of cyanobacteria and blue-green algae that can fix atmospheric nitrogen. And so that gives them a competitive advantage over the other types of algae. Next slide. So in a system where you have adequate nitrogen in the water column, so this is going to be like nitrate or ammonia, those are the general forms, then the green algae and the beneficial, the real algae, they have a fighting chance, and they're usually a little bit more efficient than the cyanobacteria. Whereas if you don't have enough nitrogen in that water column, then cyanobacteria say, great, because this is free reign for me. All my competition doesn't have enough nutrients to survive, and I can fix atmospheric nitrogen and use all of this extra phosphorus that's around. So by having a low nitrogen to phosphorus ratio, that really favors blue-green algae, whereas a high nitrogen to phosphorus ratio is going to favor the beneficial algae. So that's why I'm kind of saying, a low nitrogen to phosphorus ratio, you're just going to have this eutrophication and these algae blooms that aren't getting eaten by the food web. Whereas a high nitrogen to phosphorus ratio, you're more likely to send those nutrients up higher into the food web and grow some big bass. Next slide. And so if we think about it, the nitrogen to phosphorus ratio, since there's only two components in it, there's two ways that we could increase it. We could increase the nitrogen, or we could decrease the phosphorus. So the first aspect I want to talk about is increasing the nitrogen through a method called nitrate addition. Next slide. So in this addition of nitrate, you're really trying to accomplish two different tasks. So you can, nitrate is also a redox acceptor. And as we talked about, the redox ladder is pretty important. So it's right poised underneath oxygen. So it keeps that redox at a pretty high level. And the byproduct of denitrification is nitrogen gas. So it's inert. There's no toxic byproducts. There's no water quality contamination issues. So by having either oxygen, if you have an hypolimnetic oxygenation system like Mark were talking about, or by adding nitrate, you can poise that redox at a higher level, and you can keep it from going down into things like manganese reduction, which is bad for releasing dissolved manganese for water treatment purposes, or stop it from going into iron reduction, which is associated with phosphorus being released. Phosphorus binds really well to those iron oxides when they're solid before they get reduced. But once you have bacteria reducing these iron oxides, they form dissolved iron, and then that won't bind the phosphorus anymore. So you get phosphorus kicked up into the water column. And of course, it keeps it above sulfate reduction as well, which, they produce sulfide as a byproduct and methyl mercury. So nitrate is going to keep your enough nitrate at the sediment water interface, will keep the redox higher, and it will prevent some of these things. And then it can also be used by algae as a source of nitrogen. Next slide. So one of the case studies I want to talk about is Aquatic Reservoir over right near Washington, D.C. in Virginia. And so they had this highly treated wastewater, but it had a lot of nitrate in it because it goes through nitrification, and denitrification takes a long time. So they had, what some might consider a problem, like, okay, well, you have too much nitrate in your wastewater effluent. It's actually a good thing because they were having lower redox. So they discharged this in, and then they took a look at two different tributaries in this reservoir. So you can see it kind of winds around. But we'll look at the blue part on this little map where it says nitrate rich, and that it had much lower methyl mercury and a much lower dissolved iron and manganese, which is all indicative of a much higher redox than the other tributary, which had low nitrate and oxygen. And then you also had a lot lower blue-green algae abundance in this tributary. Next slide. And so another case study is Onondaga Lake. And this is a pretty polluted lake. It's very deep too, it's about 63 feet. So they injected calcium nitrate into the bottom water right above that sediment water interface to enhance the redox and to stop phosphorus release and to prevent methyl mercury formation. Next slide. And so you can see before that nitrate addition, so nitrate addition on this graph is, in those red boxes, that we have really low methyl mercury and soluble reactive phosphorus when you have this nitrate addition, and before that you have really high methyl mercury and phosphorus. So by adding this nitrate, we could essentially poise that redox at a higher level to stop and prevent some of this water quality contamination. Next slide. So now we'll talk about the other mechanism to increase the N to P ratio, which would be to decrease the phosphorus. And that's through a technique or a technology known as phoslock. Next slide. And so phoslock is clay with lanthanum embedded inside it. So usually clay, there's like layers of clay and they're held together loosely by like things like sodium or calcium or potassium or other metals. And so lanthanum is a natural transition metal, so it's a little more rare, but it's pretty non-toxic and it has an incredibly high binding capacity for phosphate. So whenever it comes in contact with phosphate, it will form this mineral that's completely insoluble and it's resistant to things like anoxic conditions. So it's a great way to form a barrier on your sediment water interface. And so it can prevent any of the phosphorus that's being released from being emitted into the water column. Next slide. So here's just a quick little demonstration or animation of what's going on. You could click to start the animation. And so phoslock gets applied, and then when the sun comes out, you'll see that as anaerobic respiration goes on, the phosphorus gets released and turns into algae. But the phoslock provides the barrier that creates LAP, which is rhabdophane or lanthanum phosphate, rather than allowing it to go up into the photic zone. And then the next slide. So with this one, we're stabilizing the redox with phoslock. So you can click to start it. So after we grew that initial algae bloom, then if you click once, it should start. So yeah, then some of that algae that didn't get eaten, it decays and it goes down to the sediment water interface where it drives that redox down, and you have things like ion reduction creating another algae bloom by releasing that phosphorus, and you have some sulfide being emitted when that sulfate reduction goes on, and then you finally get to methanogenic conditions and more of that water quality problems. Whereas the phoslock will stabilize that redox because you never had that initial algae bloom, and so you're keeping your sediment water interface with less organic matter. Next slide. So the first case study was a really cool, interesting study done over at UC Davis. They did a lot of work. This was just a very small part of it. So I'd say everyone go check out the poster by Nick Framstead. So it was a sediment core incubation where they did a control versus a phoslock treated. They did them under the same conditions for 15 days, and they saw that phoslock just dramatically reduced the amount of phosphorus in the water column. Next slide. And so this was an irrigation pond in North Carolina. They had a ton of blue-green algae. So as you can see, the blue bar, which is cyanobacteria, is almost as big as the green bar, which is the total algae. So it's 90% of the total algae. And then as soon as they added phoslock, you see the N to P ratio shoots up, and then the total algae abundance goes way down, and the blue-green algae proportion of that total algae also goes way down. So it was pretty successful in that aspect. Next slide. And Laguna Niguel is another case study. It's a pretty large lake down in Orange County. They added some phoslock to reduce the sediment phosphorus release. Next slide. And they had really amazing results. It's very similar. Next slide. So as you can see, cyanophyta is cyanobacteria. And so that was, dominant algae abundance before the application, whereas you totally reduce that algae and then after that application. And the next slide. And so there's just a quick summary, and I welcome any questions. So thank you guys for your attention.
Great. Thanks, Brian. We had one question in the Slack that said, when would it be appropriate to use flocks and sorbents versus redox control for nutrient control?
Yeah, that's a great question. So there's actually a term called flock and lock. So you'll flock, which is like settling out particles or forming flocks with all of the algae that's up at the surface water, and you'll take them down to the sediment water interface. And then lock is that instead of allowing them to release some of the phosphorus that's bound up in that organic matter when they decompose at the sediment water interface, then you're going to have that phoslock there to be there to absorb that phosphorus and turn it into a mineral rather than letting it go up. So that would probably be, that's a really cool, more novel use of it, because one of the things about phoslock is that it takes time to see this improvement. So it's a long-term strategy rather than some of the other techniques like something like alum, where you see improved water quality immediately. So it's kind of a way to get at both of those aspects, to improve your water quality immediately and to have those long-term reductions in eutrophic status and algae abundance.
Great, thank you. And then we had another question asking if there's a lower threshold for phosphorus beyond which N to P ratio control on the community shifts stops being relevant or effective.
I mean, it probably wouldn't be effective at a level like Lake Tahoe, where you're so deprived of nutrients that there's other mechanisms going on. So some of the algae are going to have to go for some type of an organic matter bound phosphorus or some phosphorus diesters. So when your system gets so limited, then the biota will find a way to get whatever they can, even like degrading algae at the surface. They will find a way to take that phosphorus. So it wouldn't be a mechanism that you could necessarily change with that. But in those systems, you're already usually at such a high level of water quality and clarity to begin with.
Great, thank you. All right, wonderful. Well, those are the questions, and we're out of time for that. So thank you, Brian, and I encourage everybody to turn to the Slack channel for additional questions. And want to thank all the speakers, Brian, Karen, and Mark for those great talks. And I will pass it back to Ali for the next part of our agenda.
Great. Thank you, Keith, and thank you all for those excellent talks. At this point, we are all done for the actual talks part of CABW Cal SFS. For those that want to tune back in around 1:20, you could technically go in now, but check out those first virtual posters. And then at 2:15 is the last R workshop. Again, you would have had to sign up for that. So those that did sign up and register, be back into the agenda at 2:15 to start that workshop. And thank you all again. It's been an excellent and successful CABW all virtual 2020, and appreciate all your time. Thanks.
The recording has stopped.